Could we get everybody in, please? Everyone on the outside? Well, good morning. You don't look too moggy, a little bit, but yeah. The ones that stayed the longest look the freshest. Well, I'm going to start. We don't want to go into stretching out the day. It's a real pleasure for me to introduce Jackie Tucker to you. We're shifting gears now. She's also she's on council as well, so I want to congratulate her for that, and you can now see what a council member looks like. But we're going to shift gears from animal science to the animal scientist. And I've seen a golden thread throughout the Congress about soft skills. Now, we know that our university programs are far too full to still try and push in extra things, but soft skills are incredibly important, because if we're to start relating facts in an emotive way, we need skills that we don't have, because clearly the community and the people out there are not just listening to our facts. They want to be shifted emotionally, and that requires skills other than the facts that we have and that we get at university level. Of course, you cannot emotivize a fact that you don't have. So that's why we do go to university. But these skills are incredibly important, and over the years, and Jackie's been one of those people, we've really noticed there's a need for that niche. So, and further education in that area. So she's pioneering something, and I'm really pleased about it. So Jackie, without further ado, please come and give us your best.
No pressure, yeah. Thanks very much, Paul. Thank you very much. Let's get that. Okay, there it is. So I wanted to start off this morning and say that exactly as Paul has mentioned, there have been little highlights all through the speakers speaking about how we need to address our animal scientist in the room, looking at skills development, looking at training, and we've spoken about environment, we've spoken about sustainable animal production, we've spoken about economics a little bit. But in this room today, I don't see any animals. I see people. And I think we forget about that as animal scientists. We forget that we're people. And we need to learn how to work with people. And we need to understand how they think. And we've got to be able to work with a diverse team to be able to make the differences that we want to make in the next 100 years or whatever it is going to be. And it's up to the young people in the industry to be able to really understand what is needed in this industry to go forward. So it's been really nice that there have been speakers that have highlighted that we need to have something actually change. Well, that goes very quickly. It's not just everyone else. It really does go quickly. The other comment I wanted to make before I start is that if you have a look at the theme of the Congress, it's about how we change the paradigms. How do we create a new reality for animal science? So I think it really is something. I went to Trevor a little while ago and I said to him, listen, I think this fits quite well in. And, we always speak about the science-based application of our degrees and of our professions, but we don't talk about the humanity side of it. And we don't speak about the human side. So I think it is really an important topic, and I'm really excited about what this could bring to the industry. And the reason why I want to talk about it is that all of us play a huge role in all of the food production systems. We all play a role in that. And we can have a significant impact. We already do. We have individuals in this room that have had huge impacts in the animal production industry and the whole the value chain of agriculture. And how do you as the next generation actually have that impact, if not bigger? So everything that's on our plates that we eat, and often we eat a meal, however, we don't look at it as we should as animal scientists. So the origin of our food, how has that food ended up on the plate? A lot of us are more aware of it, but we need to be communicating this. And I think it really, really is important. And at the same time, we have to pay attention to the environmental aspects. We have to pay attention to areas, as we've spoken about with climate change, etc. However, there are the society issues that we need to address, and it requires a different mindset from animal scientists to actually address those challenges in society. If we look at this, it all starts with education. It all starts at the basis of our degrees, is the education of our society. However, at the same time, it's also education for ourselves as animal scientists and understanding what's required in the industry. Education leads to healthy, productive businesses, obviously leads to nutritious food on their plates, leads to prosperous businesses that create employment, which we all know is a challenge in this industry or in the country. It also assists in the community growth. It helps them grow stronger, and at the same time, we create a space where those communities grow stronger and they actually start supporting the agricultural industry. So it really is important to look at it as a system, which has also been mentioned already by previous speakers. I wanted to go into a little bit of where my idea has originated from. So I run a business called the Keystone Collection, and I wanted to start here and explain to you what a keystone is. If you haven't heard about a keystone before, in architecture there's an arch, and the stone at the top of the arch is called a keystone. So it is the one stone that holds everything together. It's the stability in that arch that helps everything move forward. So it is important to have a look at it like that. In nature, all of us work in nature, whether you like it or not. But all of us work in there, and there are species and there are processes in nature that are also keystone in their ecosystems, and that are there to actually maintain the balance of that ecosystem, and it is an indication of how significant they actually become in their ecosystem. So we have to keep that in mind as we go forward as animal scientists. How do we become keystone in our environment, in our industry, in our organizations, and in this association as a whole? Interestingly, humans are not keystone species, if any of you thought we were, but we're not. We're actually parasites in this world, if you have a look at what we're doing to it. But how do we change that mindset? Because only we have the ability to do that. And as a team, I really believe we can. And I just have this example up here of a few species in nature that are keystone species. And all of them play a significant role in their environments. So here we stand this morning, and we've spoken about, as I've said, the environment, we've spoken about sustainable animal production, how do we confront a new reality? But we have a problem, which is, do us as animal scientists understand how to work with people? Do we understand exactly what's required from us as a team of people to address the challenges that lie ahead of us? And I really like this. I learned this a little while ago. I was on a walk in the bush, and the guy that was with us actually used this as an indication. So in nature, there are indications everywhere, or ideas everywhere for us to have a look at. So on this branch are two thorns. On the one side, it's a problem, because if you step on it, you're going to get a really bad splinter. But on exactly the same problem is the solution on the other side, because you have a needle to remove the splinter that you've just stood on. So often, if we have a look at things, inside the problem we assume is there is the solution. But it's how we look at it, it's how we create a team of people around us to identify exactly what that solution is. Because all of us in this room are a hugely diverse team, and in this problem that we sit with as animal scientists is the solution. So I think it's a really interesting thing. I use it all the time in those thoughts. So every time I see them now, I see problems and solutions everywhere. I wanted to go back to a career in animal science, and I hope everyone understands exactly what you are. And I think it's important for the young people in the room. And if we look at the definition of an animal scientist, it is a person that applies the principles of the biological, physical, and social sciences to the opportunities and challenges associated with livestock production and management. And if you have a look at these topics, it's areas that we address on a regular basis. All of the posters here, all of the short papers, all address these topics as animal scientists. I haven't seen any papers addressing us as animal scientists. And I think it is an area and an opportunity for us to go forward with as to how we communicate exactly what the challenges are and opportunities for us in this industry. So a little bit of, so that's a little bit of background. A little bit more, so all of us know that animal science is a STEM skill, so it forms part of that integrated area of study. STEM is the science, technology, engineering, and maths. And all of us approach that way to our teaching, to our learnings. And agriculture is the backbone of our society. We've heard from numerous speakers this week that it's the commercial livestock industry contributes almost 50% to the gross value of agricultural production. So half of our agricultural production is in this room. Half of what we contribute to our country is up to us. And we have to play a role in the alleviation of poverty and obviously the food security, which has already been spoken about this week. There are a huge amount of qualified animal scientists that are educated for commercial agriculture. However, we are not looking at the human dimensions of an animal scientist. There's some research, if we have a look at the literature out there, indications that agricultural students feel that they lack skills in speaking around the business skills, the soft skills, so innovation, problem solving, and also communication skills. And if I have a look at what happened last night at the debates, it is a little bit of a lack of confidence. However, these guys were an indication of how much extra potential there is in the industry, because they were confident to sit up here and to stand here and actually debate a topic that they were hugely passionate about. So it is there, but how do we get everyone to that point? There's also an indication that more than half of agricultural undergrads actually go on to study other degrees or other diplomas and certificates to improve their skill set as animal scientists and improve the opportunities they have for the career. So we need an approach that is more multidisciplinary, that addresses the challenges we have in the modern day, and we need to look at those to find the solutions to the grand opportunities, I'd rather call them, that we have as an industry, and to improve economic development. So the skill set that you have currently is you have developed or are developing as a student specialist knowledge in the areas of animal science, and we speak about the biology of animals, the physiology, reproduction, animal welfare, behavior, conservation, animal health. All those things are specialist knowledge skills that we develop as animal scientists. The degree also equips you with a range of practical application and obviously the academic side of it to be able to write scientifically and understand what you're doing. However, employers in the industry require and value skills that we don't learn. So the ability to explain ideas that are very complex, observation skills, reflection skills. So if we have a piece of research, are we really understanding the impact of that? Is it the right research that we've done? Obviously, report writing, we're learning statistical planning, independent working. How do we collaborate with other people? How do we manage our time? It's been an interesting one, and I'm probably not going to manage time very well this morning, but I think it's an important topic. And then we also need to learn how to communicate properly and how to present. So there are skills in the industry that are required, which are really important to focus on. So the animal science curricula, obviously we know that it's structured to incorporate the entire value chain of livestock. You're learning about poultry production, ruminants, all those things. And it's evident that the traditional teaching and learning of animal science students can't be replaced. We need the basic skills to understand exactly what we're doing. However, there is a need to improve the capabilities that we have as animal scientists. And we've already heard about how we're going to be involved in having to upskill ourselves on the technology side. So how do we work with big data? How do we work with software programs as animal scientists? All that stuff has been written about. And then I really like this quote that's up here, is if we have a look at the modern animal scientist, is required to have a broader technical skill than the traditional academic research and specialities that we focus on. And it is important to consider the business-related skills that we aren't looking at, especially in the dynamic environment that we're operating in the 21st century. And if you have a look at business leaders inside organizations, all of them understand that we have to be agile and we have to be extremely focused to allow the adaptation that needs to happen very quickly in the industry. So if you enter the industry right now, there's a lot going on. It's very fast-paced. You have to be able to adapt all the time. You have to understand decision-making in order to sustain the long-term business performance. And I will also say to you that agricultural-based organizations are no different to any other industry. All of them require an individual that is able to be agile and adapt themselves very quickly to any environments. So how do us as role players in the value chain, obviously we're all animal scientists in the room, however there's an industry outside there, how do we develop mechanisms to increase the efficiency of production of our graduates? So this is an input-output system. How do we improve the efficiency of our inputs, obviously you, the graduates, to the consumption, which is the agricultural industry? And it obviously requires a huge amount of collaboration. It requires a huge amount of support from everybody to understand this. And we need people that are dynamic, that are skilled, that are capable, and probably most important, optimistic about the future of the agricultural industry. So our challenge in this country, and all of us are hugely aware of this, is we need to retain those skills. How do we not lose them? How do we keep them in this country? I mean, you heard from Deerwold yesterday about how much we have to offer the world. How do we retain the animal science skills in this country? But how do we develop the skills that are required for the 21st century animal scientist earlier on? Because I don't believe that we have the time to wait for 8 or 10 years experience in the industry to actually develop those skills that are required out of a business-related point of view. I think we need to be teaching them earlier on. And if we look at the skills that I'm talking about, so on the bottom of the T, here's an indication out of a paper where they speak about the T-shaped skills. And over here is your skill as an animal scientist. We have a huge amount of depth of a speciality field that we're able to understand. But do we have the breadth of experience, of skills, and of specific knowledge around other dimensions of our careers that we aren't addressing. And how do we develop this earlier on? And it's these skills that lead to the economic development, that lead to opportunities as entrepreneurs in the industry and actually creative people to be able to develop these T-shaped skills. So it is a combination of our degree knowledge and our application of that degree knowledge, but it's also really important to develop the soft skills around the human side of our businesses. And the definition of that is a dynamic capability. So if we have a look at the definition of that, it's the ability to integrate, to build, to reconfigure internal and external competencies to address an environment that changes rapidly. There's an increased demand for sustainable food security. All of us know about that. But it offers a huge opportunity for a creative animal scientist who can work and communicate across disciplines. We also heard from a speaker yesterday speaking about we need a multidisciplinary approach. We have to look at it as a system. And in that system are people. We can't forget about that. And it is envisioned that animal scientists will play an important role in helping other scientists address these grand challenges that exist in the environment. So how do we work together with the geologists, with the engineers, with the veterinarians? We have to look at it as a multidisciplinary address. And of course, all of that goes around skills on how you work with people and an awareness of the humanity and the ethics around that. These are just a few of the soft skills up on the screen. I'm not going to go through all of them, but I've spoken a little bit about it. And we heard also from a speaker about the opportunity for leadership. And we have to develop leaders in the industry earlier on who have a responsibility and who are able to create an environment around teamwork. It really is important. And a speaker also mentioned that we have to be able to think critically. Exactly what is critical thinking? We have to question everything. We have to ask why, how, what, where, and when. Everything you do has to be questioned like that. And also remember, you don't know it all. You need a diverse team of people, and it is really encouraging to have a look at this room from up here to see the diversity that exists in animal science, and we have to utilize that. We need the diversity. And we've got to be open to others' opinions and evaluate all options. And I use a word that I've created, which is how do we get to creative collaboration? Because it is about learning from each other. It is about the opinions that we take. I have undertaken a project in my MBA dissertation. So please look out for a survey which will be going out. But it is an exploration of the opportunities and the challenges for animal scientists that address these dynamic capabilities. So I'd like to identify exactly what the skill shortage is and how do we put together a program that actually addresses that, because it is a unique industry. It is an industry that requires different thinking and actually requires a creative collaboration. The objectives of this research specifically is that higher education institutions and organizations, I'm speaking to industry here as well, need to respond to the changes of these opportunities that exist, and we've got to provide quality education for animal scientists to obviously meet the demand that exists in agricultural production in the future. However, we need to know exactly what that means to the industry and to academia. I'll skip through that. So, obviously my idea is to become a critical voice in that part and to be part of the solution that we are able to develop these skills earlier on in the classrooms and ultimately help you make significant contributions in the future. I'll skip through that. I'm running out of time. I see Paul's got his flag up. So if we go into, I'm going to go back to the keystone thing now, because I think it's really important. However, all of us have learned things over the years and early years, if we have a look at the room. But we have to sometimes unlearn something. And we have to relearn something new. So how do we reimagine the future? And I want to go to the aardvark quickly. He is a keystone species in nature, and it's the only species of its kind in its order, which is really unique. And they are considered keystone species, like I said, and are Africa's ecosystem engineers. So if we didn't have aardvarks in our ecosystems, there wouldn't be warthogs, there wouldn't be wild dogs, there wouldn't be all of those other species that use these underground burrows to either breed or nest or as a secure place. So it is a really key species for our ecosystem. And I like to look at it, that the aardvark is able to regenerate possibility and is unique like everyone in this room. So how do we develop an aardvark attitude as animal scientists? It requires us having a look at the environment that we operate in. How do we impact everything and everyone around us to regenerate the opportunities that are there? And there are all sorts of things inside nature, which is where we're working, that help us remember how to integrate and how to actually create a life that is full of opportunities in the future. And also remember, in these keystone arches are individuals or industries or organizations that can impact a larger ecosystem. And they all play a role together. So you in this room and your industry and the farms that you operate on and your organizations are all part of the food ecosystem. But how are we creating the awareness of how we integrate into that system as human beings? So I believe that us as animal scientists are a keystone species, even though I said humans are not, but animal scientists are. So how do we create an opportunity to develop and strengthen the areas around how we work as a team of animal scientists to improve our career opportunities, to develop skills that will help us in the next century address these things that ultimately nourish our world? So there's a termite mound. I hope you all have had the opportunity to see them. But it is the bigger picture, because if we have diverse thinking, integration and teamwork, our impact can be really, really significant. And if you have a look at these things, I mean, even underground, it's even more glorious. How do we do that? So if we have a look at the complexity of organizations and of a business, it requires individuals who create, integrate, and nourish people into our organizations. The industry needs to invest in improving these capabilities and soft skills for the careers of animal scientists as we progress through the 21st century and help to supply food in the future. It is, at the end of the day, about people. Thank you.
Thank you very much, Jackie. Just could we have a mic here just for one question before we move on? You did well there, Jackie. Really good. Anyone have a question for Jackie? While we're waiting for you to think, Eddie's got a question while the microphone there. All the people from UKZN, the small university of UKZN, they'd like you just to meet at tea time for a photo, and then the organizing committee too. Everyone who's involved with organizing, just come for a photo. Eddie? You want this one?
Jackie, thank you for a very interesting presentation. I think this is long overdue. It's important to focus on the animal scientist, but one of the things you mentioned, I think that needs attention as well is, and I would like to hear your comment on it. We're interacting with a number of other professions, closest collaborators would be other fields in agricultural sciences and our veterinary colleagues. But the problems we have, the reason we don't collaborate, in my opinion, is the blame society. And it's turned into a blame and shame society. So everything animal agriculture does is blamed and shamed in terms of environmental impact and so on, instead of trying to make inroads into solving those problems. And the second issue, in my opinion, is that there's no reality. It's only perceptions. So maybe we should focus on changing perceptions, and the perceptions of starting with animal scientists. I don't know how it fits in, but I would like to hear your comment.
Working now, yeah. Prof, I think it's a very good comment, and I obviously addressed this specifically to animal scientists in this talk, but I would like to address the agricultural industry as a whole, because I really believe that, and especially veterinarians, I mean, I've worked very closely with veterinarians as an animal scientist. I've made an effort to work very closely with the veterinarians over the years that I was in industry, because there's huge value in it, and it's up to us to open that communication with them and to be willing to work together. So yes, it's across industries. I'd like to focus on the whole of the agricultural value chain. I think we have to understand that it is multidisciplinary, and that environmental challenges, the sustainable food production, is not just animal scientists. It's everyone in the value chain. So I agree with you 100%. My aim is to work with the vets next. So I've already got hold of Madeleine.
Thanks, Jackie. We really appreciate that. Can we give her a hand then? Just two announcements quickly. The first one is the education forum that Haylette was going to do. That's been postponed to an online system at some stage. Haylette wants to say something. No, you're not allowed to.
Hi, everyone. I am here today if you want to chat with me, but Prof. Van Malicosta, she wants to participate and she can't be here, and because we don't have an online or virtual setup. So next week, watch your inboxes. You are going to get an invite to a Zoom, or which social media tool do you use the most? Teams. Okay. So I'll send a link via our secretary, and then please, I'm going to come prepared to the session. It's going to be good. I want our council to do what we can for you, and it's linking pretty much into what Jackie is going to do in future. So please participate, please join us, because you are our industry going forward. So we need your inputs. We need to know what we can do better. Okay, thanks.
Now that was an emotive appeal. Careful now. Then the second thing is posters are on your apps and outside there.
If you don't know which session you're on, there are lists outside. You can just find your session there, or you can look on your app. Because the posters are all on the app under the sessions.
So we have the poster session now. Thank you. I'm done.
All right, let's try this thing again. Welcome to the breeding and physiology session of the posters of 2022 SASAS. I have already explained to you how it's going to work. There will be a timer on, so as soon as you start talking, there will be a 2-minute interval during which you can defend whatever is on the screen. Unfortunately, we are not able to go according to the rankings that you have received outside. We will just go with what is on the screen. And as they upload it, we will get back to the previous speakers. All right. So the first speaker then is T. Capera. Please come forward very fast so that your 2 minutes can start. Thank you.
Hello, can you hear me? Okay. Good morning, everyone. My name is Theopoline Amushenje is my surname, Ne Itenge. So I'm also called Itenge. So I'm going to present to you research work which was done by one of my BSc honor student, Ms. Chichi Kapere, whom I supervised. I'm from the University of Namibia. So the title of the paper was Genetic Diversity of the CAP 1.1 gene in half-sheep families of Suakara sheep at Gera Post Research Station in Namibia. And I should also mention that Suakara is mainly kept for pelt production in Namibia, and it used to be called Karakul. So what was the main objective for this paper? The key objective was to be able to identify genetic variation at the CAP 1.1 gene. So that was the main objective. So if we can go to the next slide, please. Next slide, thank you. There are 2 slides. Next slide, thank you. Okay, so what were the results which we found? We found 3 alleles at the CAP 1.1 gene, and these we named as A, B, and C. And so the B allele was the most frequent, frequency of 0.61 there, and the A allele was the least frequent, just of 0.03. So why did we do this study? We really wanted to just see genetic variation so that we can try to find genetic markers which affect pelt quality. And so genetic variation at this gene of CAP 1.1 may actually affect pelt quality. So we recommend that these alleles be sequenced in the future so that association study can be done to see whether this genetic variation may actually affect pelt quality in Suakara sheep. Thank you very much.
Thank you. Short and sweet. Let us just see who's I've got on next. See, you could have talked a little bit more while we wait. I got no idea who this is because the speaker is not on there. It's her. She will introduce herself just now.
Good morning, everyone. My name is Rupumera Success Mabunda. I'll be presenting this study, Evaluation of Non-Genetic Factors Associated with Temperament in the South African Bulldog breed. My aim was to evaluate the environmental or non-genetic factors that are associated with temperament in the Bulldog breed. Yes, the Bulldog is indigenous to South Africa and it plays an important role in safeguarding South African homes and farms primarily against stock theft. So because the Bulldog is bred for safeguarding, so the temperament is a breeding objective to improve working dogs and also to improve poor welfare dogs. And again, determining these non-genetic factors will help in facilitating genetic improvement in the Bulldog breed. So looking at the results of this study, we found that all the non-genetic factors investigated in this study were influenced the temperament of the Bulldog breed, where we found that in sex, females were more obedient, reliable, and fearless when compared to males. And also, as these dogs age, they were less obedient, reliable, and also they were less fearless. And for coat colors, we found that the black and the brown ones, they were more reliable and fearless when compared to the fawn and the reddish brown dogs. And also for the season that these dogs were inspected for this temperament, we found that the summer and the autumn ones, they were more reliable compared to the winter. So that in conclusion, we found that these non-genetic factors were all having influence in these temperament traits. So I can recommend that these puppies should be trained early and be given socialization training, and also they should be given a good environment for them, because when they are older, they are difficult to train, and also they were reported to have less reasoning skills, and they are less desirable to wanting to be involved in activities or wanting to play. So also even the coat color. Thank you so much.
It seems like people are really taking me seriously when I said I will forcibly remove you. Next poster, please.
Thank you. Good morning, everyone. I am Miss Nozipo Makakula, and I will just be giving a quick overview on the project titled Parentage Verification Using SNP Genotype Data in South African Bonsmara Cattle Breeds. So now the aim of the project was to validate pedigree records using SNP genotype data, as the frequent problem that is faced in smallholder communal farming systems is that most pedigree records seem to be inconclusive or absent, and therefore, can you please go to the second slide? And therefore, upon investigating that, we had a parental three-sire generation line on which the SNP genotype data showed to have more half-sibling relationships and was incongruent with our pedigree records, which then could be recommended to use SNP genotype data in cases of inconclusive pedigree records. So SNP genotype data can be used to validate pedigree records. If anyone has any questions, you can ask. Thank you.
Thank you. Let's leave the questions until a little bit later. Thank you. Next, please. Technology is good, but sometimes not so much.
Show you the slide like that.
We have that at all. Right. Machete, are you here? Miss Machete. That was really a quick talk, so quick we didn't even hear it.
Good morning, everyone. Okay, my name is Jo Matalele. I'm going to present breeding practices of indigenous goats, farmers in the rural communities of Limpopo province. The objectives of the study were to identify existing breeding practices and breed management constraints encountered in indigenous goat population. Next slide, please. Okay. The flock composition result reveals that the mature Jews, followed by the young females and the kids, represented the largest class of proportion, while the males, the castrated, the young bucks, and the mature bucks represented the lowest proportion. In terms of selection criteria, growth rate, followed by shape, color, size, and fertility were perceived as the important traits for selecting breeding bucks, while fertility, followed by growth rate, shape, color, and size were the important traits for selecting breeding groups. When it comes to culling criteria, body weight, followed by size, shape, color, and poor fertility were perceived as the important traits for culling the breeding males, while poor fertility, followed by old age and shape, were the predominant culling reasons for the breeding females. Most of the former practiced uncontrolled mating, and the males were castrated at the age of 6 to 12 months to control breeding and for better temperament. Constraints to the breeding management were mainly the lack of secure grazing land, diseases, and breeders. In conclusion, the breeding practices and breeding management constraints revealed in this study could be instrumental in making informed decisions on management, breeding, and conservation.
Thank you very much. While we're waiting for it to come forward, the speakers, please, if you turn your head to look at the screen, please turn the microphone with you. Don't leave the microphone here and turn your head and speak there, please.
Good morning, everyone. My name is Bonso Matemani, and the title of my study is Genetic Parameter Estimates for Lactose Content and its Relationship with Other Milk Production Traits in South African Holstein cattle. While lactose is an important milk component and has gained economic interest recently, the objective of the study was to estimate heritability and repeatability of lactose in the first 3 parities and to estimate genetic correlations of lactose contents with other milk production traits. Heritability estimates were moderate within parities, indicating that selection for lactose will be expected. Repeatability estimates were moderate, demonstrating that early lactation records can be used can fairly predict the performance of an animal later in lactations. Genetic correlations within parity were moderate to high, suggesting that this trait in different lactation can be considered as the same trait, especially in later lactations. Genetic correlations between traits within parity for milk yield and lactose percent were high and positive, suggesting that a possible desirable genetic correlation as opposed with other milk production traits. In conclusion, lactose percent appeared to be moderately heritable. Therefore, improvement through selection is possible. Selection for lactose percent may be based on first 3 lactation records. The desirable genetic correlations of milk yield and lactose percent within parities improve accuracy of selection in a multi-trait analysis in the South African Holstein cattle. Thank you.
Thank you very much. Next, please.
There's a recording now, Helena.
Right, speaker, please. Come, run. I'm just kidding.
Good morning, everyone. I'm going to present to you Morning. I'm going to present to you my topic on runs of homozygosity identify QTLs in genes associated with semen traits in South African and Chinese bulls. The aim of the study was to use genome-wide runs of homozygosity to identify genomic regions that are associated to semen QTLs. Oh my gosh. Our results, approximately 7,516 runs were observed between classes 0 to 5, with the most runs showing in Nguni Angus and Chinese Simmentala. 10 genomic regions had been identified with QTLs associated on BTA6, with the greatest coverage of SNPs in Simmentala. Gosh, this is a typing error, sorry. With the greatest coverage of SNPs in RAN on BTA6, showing Simmentala, revealing suggestive QTL for sperm motility, sperm head abnormalities, sperm tail abnormalities, and revealing genes MACH1 and WDR19. Chinese, Belgian blue, and South African Angus revealed the same genes. What happened here? Okay, in conclusion, the study identified distribution of runs in 6 selected Chinese and South African beef cattle breeds using genomic markers, and also showed that Angus, Nguni, and Chinese Simmentala had the highest frequency of length of runs between classes 0 to 5, indicating higher recent inbreeding in all the breeds, including Simmentala. Thank you.
Thanks, guys. It's really challenging for people to present a whole research topic in 2 minutes. So if you do need more information on the topic, these posters have been put up on the app, and it is also available on the website if you want to read more about it.
Greetings, everyone. My name is Ntobisi Sakile Ngobo. I'm from University of Zululand. I'll be presenting under the topic says the effects of cellular supplementation on semen quality parameters and scotal circumference of Zulu rams. These were my objective. My first objective was to evaluate the effects of cellular supplementation on semen quality parameters of Zulu rams. The second objective was to evaluate the effects of cellular supplementation on scotal circumference of Zulu ram. Moving on to the next slide. These were my findings. The first graph, figure 1, represents the effects of cellular supplementation on semen pH and appearance of Zulu sheep. The second graph represents the effects of cellular supplementation on sperm concentration and semen volume of Zulu sheep. The third graph represents the effects of cellular supplementation on sperm motility, morphology, and viability of Zulu sheep. And the fourth graph represents the effects of cellular supplementation on scotal circumference and statistical measurement of Zulu sheep. In conclusion, sperm appearance and semen volume were significant between the treatment groups. Semen pH, sperm motility, sperm concentration, sperm viability, morphology, and scotal circumference were not significant between the treatment group. In conclusion, cellular supplementation do not affect scotal circumference and most semen quality parameters except for semen appearance and volume. However, there is a need to conduct more study on different levels of cellular dosage, since Zulu sheep are hardy breed and may require different dosage as opposed to exotic breed. Thank you so much.
Thank you. This is Georgette. She's going to talk to us about Nguni breeds and methane production.
Good morning, everyone. My name is Georgette Pius from the Agricultural Research Council. Today I'll be talking about the effects of crossbreeding on methane efficiency in beef cattle. So the main aim of this short preliminary study was to measure methane efficiency of different beef cattle genotypes. So we took kgs of methane over a kilogram of calf weaning weights, and we used this data from the crossbreeding project that has been carried out at the Far Lots Research Station. Next slide, please. So if we look at the results, the 205-day weight and the methane efficiency of the purebred calves averaged 198 kgs and 1.69 respectively. The average heterosis attributable to the crossbred calf was that of 2% for both traits. The Nguni was most efficient among the purebred calves and had greater maternal additive effects compared to that of the Afrikaner and the Bonsmara. The greater efficiency of the Nguni dams, however, resulted from breed complementarity, where small cows were mated with sires of greater genetic potential for growth. These results, in conclusion, indicate that crossbreeding can have a small to moderate effect on weaner calf production, with most of the benefit coming from proper use of breed additive effects. So this information can, in the future, possibly be used to develop a model to estimate methane efficiency for different breed types of in-beef cattle. And also it can be valuable in determining which breeds are more well-suited for beef production, and also given the higher methane efficiencies and popular demand for lower carbon footprints in the beef industry. Thank you.
Thank you so much.
Good morning, everyone. My name is Mweki Eti, and I'm from ARC. I'm an intern. So my talk today is about the project that I was busy with, phenotypic characterization of indigenous chickens from Limpopo and KwaZulu-Natal. So the aim of our study was to define or to describe the phenotypic traits of indigenous chickens based in Limpopo and KwaZulu-Natal as a baseline study for a subsequent or a future genome-wide association study to investigate, genomic regions that could be associated or that could contain candidate genes that contribute towards the expression of certain phenotypes. So, if we could go to the next slide, please. So we observed quite a lot of phenotypic variation, and I think this was expected, with village chickens. And then what we also saw is that our variation was across the districts. We had 4 districts in our study. It was across districts and across the 2 provinces. So in other words, each district almost had a unique prevalent phenotype that you could identify. We also measured body weight between the 4 provinces and the 4 districts, and we found that our Limpopo chickens weighed significantly less than KZN chickens, which could be worth investigating. Another thing that dictates, phenotypes is farmer preferences, market demands, things like cultural roles that the chickens play. So these things also play a role in determining what kind of phenotypes are seen more often than others. There were some phenotypes that were more prevalent in some. Okay, thanks, it's fine.
Very interesting study. Thank you for that. And I'm sure there will be plenty of questions coming. Now we will have Ms. Masete.
Good morning. I am Masete Hutato, and I'll be presenting under the topic, the phenotypic characterization of the Bapedi sheep. The Bapedi sheep are at risk of extinction like most of the indigenous breeds throughout the world, and therefore, conservation of the breed will not only prevent extinction but can be used as a model towards improving the breed. In the quest for breed improvement and conservation, characterization becomes very vital. And in this study, the Bapedi sheep was characterized in order to differentiate it amongst other breeds, and also this study was done to describe the breed. And this study was initiated to identify the phenotypic characteristics of the Bapedi sheep for conservation purposes, and it was also done to determine the correlation among the phenotypic quantitative characteristics of the breed for selection programs. Next slide, please. Okay. In terms of the qualitative characteristics of the Bapedi sheep were found to be multicolored with a white coat and a brown head. And they were also found to have glossy medium length coat cover with brown eyes being more dominant than blue. And in terms of the quantitative characteristics of the Bapedi sheep, the age of the ram did not differ significantly in the study, and it was also found that the Bapedi sheep, the rams were weighing 62.6 while the ewes were weighing 45.0. And in this study, in terms of the quantitative characteristics, the ram were more superior than the ewes, and also the surface circumference weighing was 29.2. And in terms of the Pearson correlation coefficient values amongst phenotypic quantitative characteristics of the Bapedi rams and ewes, the age of the ram was found to be significantly correlated to the body length, the body depth, body height, body weight, and headgear circumference, and rump width. And there was also a slightly higher correlation between the body weight and headgear circumference, which shows that headgear circumference can be used as a sole predictor of body weight.
All right, this is all the uploads that we have so far. We are just waiting for them to update it a little bit.
Anyone here finished present that's not staying?
Many. Anybody else with presentations that still needs to be loaded, please come forward now. All right, we will have Dr. Klute presenting a poster on behalf of Ms. Tebes, and then we are busy loading, then we will carry on with the program, please.
Yes, so I'm quickly talking about on behalf of Megan Tebes on tail length and the heritability of it in sheep. We know that breed fly strike is a trait that is of economic importance, but also of welfare importance. And tail docking is applied to curb. Closer to my mouth. Okay, so tail docking is employed to curb fly strike a bit. And so what we did is we used about 1,500 records to look at the fly strike. And what we saw is that at tail length, what we saw is tail length is heritable at about 30-odd%, but it was pretty much size dependent. But even after using live weight at docking as a covariate, still some heritable variation remains. So we contend that it might be feasible to genetically change tail length, and doing this, make it unnecessary to dock tails. So this is the story.
Thank you so much. It might be a very important welfare issue in future.
Good morning, everyone. My name is Dombizota Bovula. I will be presenting the study, Evaluation of Cattle Fertility in South African Smallholder Dairy Herd using Conformation and Physiological Analysis. Firstly, we determine the age, scrotum circumference, body condition, semen parameters, as well as testosterone concentration. Secondly, we determine oestrus response and hormone concentration of cows. Next slide. Next slide. We found that bulls had good bull breeding soundness, as it is indicated by age of 6 years, scrotum circumference of 35 centimeters, body condition scoring of 2.9, testosterone concentration of 10 nanograms per mil, and semen motility of 80. We also observed high oestrus response at 95%, which indicate that cows are suitable for breeding through oestrus synchronization and artificial insemination. We also found that progesterone concentration on day 0 was 4 and on day 10 was 1.49 nanograms per mil, whereas estradiol concentration on day 0 was 3 and on day 10 was 8.4 nanograms per mil. Low levels of progesterone indicate that the cows were on heat before artificial insemination. The increase of estradiol levels may have positive effect on fertilization. In conclusion, smallholder dairy breeding animals examined in the current study appears to be suitable for breeding through assisted reproductive technologies, as it is indicated by morphology and physiological analysis. Thank you.
Thank you so much. Eventually it's your turn. I promise you'll be second, and now you're almost second last.
Good morning, everyone. My name is Bhavini Kuverji. As you know, crossbreeding is one of the strategies to mitigate climate change. So in an experiment involving crossing a Nguni sire with a Bonsmara dam and a Bonsmara sire with a Nguni dam, the resulting F1 generation showed differences in response to climate change. Hence, our study aimed at identifying structural variants in the Nguni and Bonsmara crossbreds using whole genome sequencing. For this talk, I'll be focusing on the objective to determine the quality of the whole genome sequences that we obtained from these crossbreds for further analysis. Next slide, please. Sorry, next slide. Okay. So our crossbred sequences, we detected mapping ratios between 93 and 95%. Furthermore, the VCF file showed us revealed structural variants such as SNPs and multi-allelic sites. We had approximately 8.6 million SNPs per individual. Upon further investigation of the SNPs, we observed more transitions than transversions, and this is expected across the genome. The transition versus the transversion ratio was high across all samples. When I mean high, I mean it's high, greater than 2, and ours was ranging between 2.12 and 2.18. And this is a good indicator of the quality of sequence. And based on this, we can further use these sequences for downstream analysis, and this will include copy number variation, as this is the largest form of structural variants, and to identify specific genes in these CNV regions that are specific to the Nguni side and the Bonsmara side that will help to display the differences, especially those relating to adaptation in these two crossbreds. Thank you.
Thank you, ma'am.
Good morning, everyone. My name is Tumelo Thelma Matua. I'm presenting the title, The Effect of Permitian crop protectants on fertilization rate of pig oocytes. The objective of the study was to determine the survivability of matured oocytes after exposed to dimethyl sulfur oxide DMSO in 7.5% plus ethyl glycol 7.5 when combined on holding solution, and DMSO 15% plus ethyl glycol 15% on fertilizability following IVF on vitrification. The results of the study and discussion was there was significant difference on non-pronucleus on control, which was 24.2 plus or minus 17.2, and the combination of DMSO and EG, there was 50.7 plus or minus 21.6, and the total fertilization rate of control was higher, 75.8 plus or minus 17.2, and the combination of DMSO and EG was lower in total fertilization rate, which was 50.3 plus or minus 21.4. The results indicated that exposing oocytes to cryoprotectant after maturation induces subsequent alteration in the total fertilization rate to be lower in non-pronucleus and to be higher in total fertilization rate. In conclusion, the combination of DMSO and EG had lower rate of pronucleus since cryoprotectants have toxicity when used on pig oocytes after maturation. Thank you.
Good day, ladies and gentlemen. My name is Aleta Mahoro. I'm going to be giving a presentation about my study, Genetic Characterization of Non-Descriptive Indigenous Goat Ecotypes from South African Rural Communities. The main objectives of the study was to identify ecotypes important for conservation and to quantify the genetic contribution of each ecotype. So in the findings, over 200 animals remained for downstream analysis and 47,000 SNPs. So we also find out that in the principal component analysis displayed amongst the populations, there was 6% variation, and in the principal components number 2, there was 3% variation. Now, in the genetic parameters that were estimated per population, we're able to identify that the Free State province has the highest expected heterozygosity, while the Gauteng province has the highest observed heterozygosity. We also found out that amongst the population that were studied, even though the inbreeding was not high according to the standard of the inbreeding, but the studied population showed that our conservation flock from Agricultural Research Council had the highest inbreeding in the studied populations. So we furthermore did the AMOVA variation amongst the population. We found that there's only 2% variation amongst the population. However, the variation within individuals in overall general was higher at 92%. So the interesting part about this as population was that the non-descriptive population from the 4 provinces of South Africa had similar genetic clusters with our reference population, indigenous Vel goat, Xhosa Loop, and Mbuzi. For further questions about the structure of the reference population, you can come to me after the presentation. We also found out that our conservation flock was represented by different genetic background, which was very interesting. So in conclusion, we realized that the different production system had an effect on the levels of the studied genetic diversity, and we also realized that based on our fixation indexes, we had the highest level of genetic diversity observed, which means that our population had the healthiest genetic diversity. And in conclusion, we can say that this study serves as the basis for conservation for our indigenous goats. Thank you.
All right, we have 2 more presentations before tea time, so let's get this going quickly.
Good morning, ladies and gentlemen. My name is Fopela MD. I'll be presenting about the influence of estradiol levels on cattle oocytes fertilized using sex-sorted semen. So the aim of the study was to investigate estradiol concentrations. So those concentrations were selected in terms of we were trying to mimic the concentrations in vivo for the pre-ovulatory follicles. So we determined the fertilization rate using the sex-sorted semen and the non-sex-sorted semen. So for the results, we found that the sex-sorted semen basically had the lower fertilization rate in terms of those concentrations that we used, and as well as the cleavage rate percentage for the sex-sorted semen were found low. The reason why for the sex-sorted was low is because that in terms of the technique or the sorting technique that has been used, so it actually tampers with the sperm ability and the capability for fertilization. So in conclusion, we concluded that the presumptive zygote that were developed at these concentrations and fertilized using particularly the sex-sorted frozen without semen provided the low oocyte developmental competency as compared to the unsorted sperm. Thank you so much.
Thank you. And the last one for today in the poster session coming up.
Good morning all, I'm Sishabela LT and I'll be presenting to you assessment of MTA content on liquid stored semen of Goldbrook boars following varying glutathione concentrations. So Goldbrook is an indigenous pig breed characterized with extreme adaptability traits which include disease tolerance and the ability to survive under harsh conditions. So the aim of the study was to determine the effects of glutathione concentrations, supplementation in boar semen extender on the MTA content of liquid stored Goldbrook semen. Next slide. The results were semen treated with 10 millimolar of glutathione showed the lowest MTA content as compared to the other treatments at 3 hours. And then at 6 hours, 5 millimolar of glutathione showed the lowest MTA content as compared to other treatments. And then meanwhile, there were no significant difference among the 1 and 5 millimolar of glutathione. So these results indicate that as the sperm cells were able to maintain their survival and were able to survive at a low lipid peroxidation due to the activated antioxidant defense system. In conclusion, liquid preserved boar semen combined with the various glutathione concentrations at lowered MTA content at 6 and 24 hours were able to maintain the lowered MTA content. However, 5 millimolar was the optimum concentration to be added to the boar semen extender during the liquid preservation of Goldbrook boars. Thank you.
Thank you very much, ladies and gentlemen. From a personal viewpoint, I really want to commend all the speakers in this poster session for sticking to the time so well. This is really a difficult thing to do, and I think you all did a sterling job. Congratulations. I think we do not really have time for questions in this session at the moment, but we are breaking for tea now. So if you do have a question for any one of the speakers, please catch them outside. And if you don't get any questions, go out for a beer so that you can relax. Thank you very much.
Oh, 7 hours driving on the highway. Far away, far away, far away. Every moment I was spending on regrets was such a waste, such a waste, such a waste. Looking on the city lights, flashing bright in my eyes. Look up to the sky and I got this relief inside. Then there's no need to hide. I'm going all the way up, up. I'm going all the way up, up. I'm letting go in my heart, heart, of everything that holds me down, no, no. And it's feeling like I'm in love. I can feel the love, oh yeah, it's all around me. Oh, I need to know how I can make it, not afraid to fall and perish it behind me. Feeling like a stranger on a foreign land, far away, far away, far away. I could get along, could be a different man. Looking on the city lights, flashing bright in my eyes. Look up to the sky and I got this relief inside. Then there's no need to hide. I'm going all the way up, up. I'm going all the way up, up. I'm letting go in my heart, heart, of everything that holds me down, no, no. And it's feeling like I'm in love. I can feel the love, oh yeah, it's all around me. Oh, I need to know how I can make it, not
let's start. Good morning, everyone. My name is Linky, and I'll be the chair for the session. So we had a very good congress so far, so I hope we wrap it up accordingly. To all the speakers, 15 minutes is all the time you have, so you can present for 30 minutes and 2 minutes of questions. And we do encourage that speakers leave some time to engage. So we'll start with the very first presentation, which is a virtual presentation by King et al. The title of the presentation is Morphological and Genetic Characterization of 3 Mozambique Indigenous Cattle Breeds. I tried to learn how to say that word, but I struggled. So over to you, King.
Good morning, everyone. I'm here to present my research on morphological and genetic characterization of 3 Mozambican indigenous cattle populations. This research will be presented in 5 sections as follows: background, aim, methodology, results and discussion, conclusions and recommendations. Indigenous cattle contribute to rural livelihoods in Mozambique by combating food insecurity. These cattle are the majority of cattle in Mozambique, and they are important animal genetic resource because of their adaptability and resistance to a number of tropical diseases. However, there is limited studies done to investigate their genetic variation and also their genetic relationship with other cattle in the region. The aim of this study was to access the phenotypic and genetic variation among indigenous cattle breeds in Mozambique. Specifically, to characterize phenotypically these breeds, to investigate their genetic diversity, to determine the population structure and administrative levels among these breeds, and finally, to compare Mozambican indigenous cattle with South African Sangha breeds. Methodology, sampling and data collection. This study was done in 4 provinces of Mozambique, namely Maputo province, Gaza and Inhambane in the south part of Mozambique, and also in Tete province, which is located in central part of Mozambique. Data collection and genotyping. Each breed was sampled where it is predominantly found. The sampling strategy consisted of identifying 3 districts per province, 2 villages per district, and 3 farmers in each village. In order to minimize related animals, sampling was done in different grazing areas, and no more than 3 animals per head were allowed. The sample size for phenotypic characterization was 140 for Angone, 292 for Landi, and 182 for Tete cattle. Body measurements were taken in adult animals and non-pregnant females. The traits measured were age at birth, muzzle circumference, body length, horn length, top line length, 8 widths, 8 ramps, ramp wide, hock circumference, and body weight. For genetic characterization, hair samples were collected from 228 animals and then were genotyped using IDB3 SNP chip. The South African genotypes were generated by TGP 8K panel and then compared with Mozambican Nguni. The sample size for South African genotypes were 150 animals for each breed. Data analysis. Phenotypic data was analyzed using SPSS version 16. Heterozygosities and the inbreeding rates were calculated using Blink, and effective population size was estimated using SNAP. Principal component analysis was performed using GCTA, while population structure and differentiation were detected using admixture and adgenet. Results and discussion. In Table 1, we show the average body measurement of cows. In this table, we can see that there were marked difference in all measurements except for muzzle circumference. The Angolan cows were lighter and smaller than other breeds, while the Landing cows were heavier than other breeds. In Table 2, where we show the result for stepwise selection of the traits studied, we can see that 6 out of 10 variables were stepwise selected. Among them, the body length and the 8 width were the most discriminant traits for the 3 breeds, and these variables, they can be used to differentiate the 3 populations. Genetic diversity parameters. Table 3, we can see that the inbreeding rates found in all 3 populations were low positive. All the inbreeding rates were positive and low. The observed heterozygosity and also the expected heterozygosity were similar in the 3 breeds, were very similar in the 3 breeds. That show marginal superiority to the genetic diversity. In Figure 2, we show the administer graph indicating the proportion of ancestral population for each individual from 3 cattle breeds. This picture indicates poor between breed differentiation. So there were no clear differentiation between the three breeds. The three breeds showed evidence of admixture and highly heterogeneous genetic background. FT population size show a downward trend in all three populations, and at generation 12, the new which is FAT population size was 627 for Angoni, 929 for Landi, and 349 for Tete. Analysis of the principal components for the four breeds, South African and the Mozambican one, three South African breeds and the Mozambican one, show 4 clusters. The Mozambican Nguni and South African Nguni population showed an overlap in clusters, while the South African Tuli and the South African Boran were the most genetically distant, with FST of 0,027. Table 4 show the raw statistics in the 4 populations. This table shows us that the inbreeding rates for all the 4 populations were very low, and there were high frequency of short rows. The largest number of rows was found in South African Tuli. These results indicate that there was an ancient inbreeding in all population. Because of this frequency of short rows. As conclusion and the recommendation, we can say that Mozambican indigenous cattle have moderate genetic variability and limited genetic differentiation. Mozambican Nguni and the South African Nguni are genetically linked and share the same ancestry. There is a need for a regional strategy for the preservation of indigenous animal genetic resource in the region. Thank you very much.
Thank you for the presentation. Colleagues, we have a minute or so for questions or comments to the presentation. Kaswa, there's a question there. We have a mic. Do we have another mic?
Thank you. My name is Dou Chokwe, and thank you, the speaker, for the good presentation. Just on your conclusion, I've noted that part of your conclusion is that there is a need for a regional strategy on preservation of indigenous animals. I just wanted to know from you if you are aware that there is a SADC strategy on conservation of animal genetic resources, which you can go and find it on any internet. Are you aware of it?
No, I was saying that no, I didn't know that there is such strategy, that this research, if there is the strategy, will be part. We can join to that strategy so that we can have these indigenous cattles conserved. But from now on, I will go and search for it.
Okay. There are no more questions. Let's give him a round of applause. Thank you for that presentation. We move on to another online presentation by Kgaugelo Mafulo, and the title of the presentation is Multi-Breed Genomic Predictions in South African Holstein and Jersey Dairy Cattle.
Hello everyone, my name is Kgaugelo. I'm working with Dr. McNeil, Prof. Neza from Akatlela. The title of my presentation is Multi-Breed Genomic Predictions in South African Holstein and Jersey Cattle. So the background of the study is that genomic prediction simply refers to the inclusion of SNP genotypes into genetic evaluation models for prediction of genomic estimated breeding values. So this is the actual model for the routine evaluations. Now they're incorporating the this DNA technology into the evaluation to evaluate the potential of an animal as a potential breeding animal as soon as the DNA is available, and this procedure is shown to have improvement in the breeding industry. So the genomic predictions using both single and multi-breed methods are currently taking place around the world, and the accuracy of genomic predictions, among others, include the size and the structure of the reference population. Reference population is the group of animals with both performance record and the SNP genotypes. So developing countries like South Africa are struggling to introduce the genomic predictions, mainly due to lack of AI balls, financial constraints to establish large reference populations. These are the studies that were taken using small reference populations, which were mostly predominantly cows, including the one that we did in South Africa, which is unpublished. And you see that the accuracy of prediction, among others, is at less than 70%, and you see the reference population is very low. And in the effort to improve the accuracy of prediction, the inclusion of cows that we see previously, where there are no AI bulls, and using multi-breed, where we are combining different breeds into one reference population, is currently explored to increase the size of the reference population. So in South Africa, we have the stat book that is currently offering genomic selection services. We just need to highlight that, but there's limited information on genomic prediction in South African Holstein and Jersey cattle around multi-breed genomic, multi-breed and multi-trait genomic predictions. So the aim of the study was to assess the accuracy of multi-breed genomic prediction using single trait and multi-trait model in South African Holstein and Jersey cattle. So the flow of the data, the study was approved by the ARC ethics committee. The data pedigree performance we received from the integers, and the genome type data was provided by the dairy genomic program DGP. And then we had this data, we have the before and the after, and then I've shown also the editing criteria that we have used is according to the Interpol standards. And then also I've shown how when we combine the data from both breeds, how it went, and the data that we used, it was for milk, protein, and fat yields in kilograms. And then also for the genotype data, we used 50K SNP genotypes with 53,218 before editing, and after editing we're left with 2,600, and we had 2,574 animals that we used for evaluations. And then this is our descriptive statistic of the performance data. Our milk yield ranged between 1,000 and 25,993, and the mean was 6,600. And then the heritability here is 0.21, and the same results are, the same statistics here are indicated for protein and fat. The modules that we used two modules, the traditional blab and the single step genomic blab, which are indicated here. So the traditional blab that we used is the repeatability model, where we used the lactation numbers as the permanent environmental effects. And then in terms of the SSG blab, we similar to the traditional blab, but the difference is that the A matrix now is replaced by the age matrix, which the age matrix is made up of the A matrix and the G matrix. The G matrix is the genomic relationship matrix by Van Reyden. And then we had three modules: the single trade, the two trade, the true trade, and the third trade. And we had two types, which is same, where the breed remains the same and the breed is considered as a fixed effect. And then our evaluation pipeline is as follows. We had this model. We predicted the ABVs for all the animals using the blab model. We removed their phenotypes, and then we estimated their EBVs and their GEBVs. And also we used their Holstein and Jersey as a reference population. And then we had three validation populations: only when there's Holstein, only when there's Jersey, only when they combined Holstein and Jersey. And then we extracted the EPVs and GEBs from of the validation animals. That means we took the EPVs from this step 1 and the EPV and GEB from this step 2, and then we correlated them using the Pearson correlation coefficient. And then the results for the PCA plot shows that these are two distinct breeds. However, in the Jersey, you see that there's a bit of genetic diversity there. And then in terms of our results, I put them into blab and the single step G blab separately, because we've got the same and we've got the diff. And then also we've got the modules here: single trade, two trade, and third trade. And in terms of the two trade, we have here is one: milk and protein are paired together, milk and fat paired together, and protein fat paired together. But from the results, we see the accuracy was very, very low. But the only difference here is that we have about 15% when we had a Holstein as a validation, and when we fixed the breed when we put the breed as a fixed effect. That's what we found when we are using the blab. But when it comes to the single step G blab, where the table is still the same, the arrangement is still the same, only the different results here. But we see with the results we are finding better accuracies, but they're still the same in terms of the validation. Holstein is still having better accuracies. And what we see here is that the single trade has up to 20%, the two trade has up to 28%, the third trade has up to 26%, but the consistency of the third trade is that you have between 20 and 26% accuracy. And what we see as well is that Holstein is performing better than when Jersey is used as validation or combination of the two, which is somehow concerning. But we comparing them with the research from other studies, we see that the results that we have are very low. So the studies, they all used Holstein and Jersey as a validation. As a validation and also both as a reference population, and using the GBLAB model. And then what we see here is that they have high accuracy compared to what we have in our study. So, and another thing that I noticed that when it is only KLAS who did the single trade and the multi trade, and found that the single trade performs better than the multi trade, or almost equally. And yeah, that's something that we found, that these results are better than what we have found, but as well they are considered as low because they say accuracy of prediction for multi-breed is low compared to single trade. And then what we are going to do going forward is using meta founders and using GRM, which I'm not going to go much into details, but the meta founders are just group of animals that serve as proxies for animals in base population. This is an upgrade from the unknown parent group, or the unknown parent group, sorry. And then the new GRM here, what we see is that the different studies that have tried to see the impact of modifying or improving the GRM, that will be like spread specific GRM that as well will improve the accuracy of genomic predictions. And also there are those that realize that when you have breeds like Holstein and Jersey that are not related, how about you set the covariances of the GRM to be 0 to set that there's no relatedness between the two breeds, because when you have a negative relationship now, it's going to affect the outputs of your genomic predictions. But so these are our preliminary studies that we found meta founders and the new GRM. We see that in the meta founders, we used the three trait model. These are the results of the three model from the previous slide. These are the results from the meta founders. We see that meta founders has improved the results, but it's still the same. Holstein still as a validation is still performing better than our Jersey, or the combination of the two. And another thing, when it's come to the new GRM, we see that when you're combining the breeds, the accuracy is better. But for Jersey it remains the same, and there's a reduction for from when you are using Holstein. So this is still an ongoing research. In our conclusion is that Holstein as a validation resulted in reasonable prediction accuracy compared to Jersey and combined of the two. And the prediction accuracy of multi-trait shows potential in multi-breed genomic evaluation compared to the single model, and accounting for breed as a fixed effect in single-step GPLAB tend to increase the accuracy of multi-breed genomic prediction. So exploring meta founders and new GRM or new genomic relationship metrics are recommended for improving the accuracy of prediction in South African Holstein and Jersey cattle multi-breed genomic predictions. I would like to acknowledge these institutions. Thank you very much.
Okay, thank you, Kgaoelo, for the presentation. Colleagues, are there any questions for the presentation? Anyone else? We still have time. Okay, so you go first and then we'll go to you.
Thank you. Oh, okay. Thank you very much for a wonderful presentation. I just wanted to have a couple of questions. I wanted to find out what is the advantage of using this SNP genotype technology, which you used in your study, and also I want to find out the cost of this SNP genotyping per sample, and when it is quite high, support is quite high. I wanted to find out what do you think is a strategy which can be used in order to make sure that farmers actually are encouraged to use this technology. And I also wanted to find out why do you think your values were much lower than those of other researchers, because your values they were actually lower. Thank you.
Okay. Kgaoelo, did you get the questions?
Yes, I got the question, Prof. Thank you very much. The advantage of genomic predictions is that you are actually shortening the generation in interval. So instead of doing traditional breeding or traditional evaluation, where you spend about 7 years, you can actually have a DNA of the animal as soon as the hair sample is available. And using the genomic selection, you can do the genomic estimated breeding values and see the potential of that animal as a breeding animal. And in terms of the cost, it is still expensive to genotype in South Africa as compared to other countries. Like if you look at the Holstein, we have about less than 3,000. We've got a lot less than 3,000. But if you look at other countries like USA, they've got more than 3 million genotypes, and for you to increase the accuracy, you need more animals genotyped and also increase the amount of relatedness as well. So in terms of making the farmers or the small holders, holder farmers adopting the technology, it's all about educating them, making them see the need for this technology, because as well you can be able to measure animals that they do not have pedigree. That is the prevalence in most smallholder animals. So that's one of the advantages with the genomic selection. So the reason why we have the low prediction accuracy is that, so here we're doing a multi-breed genomic predictions. So it has been shown that multi-breed prediction is very low because of there's a difference in the genetic architecture of different breeds. So that's one of the contributions. The reason we're doing it is because we want to increase on the number of animals we're using in the reference population, because the higher the reference population, and it's been shown that it contributes to higher prediction accuracy. So yeah, so it's all about numbers. So we just trying to leverage on that. Thank you very much.
Hello, we only have 20 seconds or something.
Okay. Thank you, sir, for the interesting presentation. I just have two questions. The first question is, what is the age of your sampling group for the cattle? Because in dairy, we know that the older cattle tend to produce more beta solids, which are your milk yield, fat, and protein. And then the second one is the generation interval of the study, because in dairy, the generation interval is 3 years, where you'll see every 3 years there'll be improvement in your genes, in your gene pool in the head. So I would like to know the age of the group and the generation interval. Thank you.
That's a very good question. In terms of the generation, in terms of the age, we did not look at the age, but we used animals that were from 1990 to 2016. Those were included in the our evaluation model, and we used the data, but we edited according to interval standards. So it might be difficult to give you that answer. So
However, we had 3 lactations. Genotype animals span from 1990 until 2016, yes, he said. But the other data were spanning for all the data that were available in the database, although we evaluated only 3 lactation. Let's move on to the next presentation. Let's give him a round of applause. Thank you. Let's move on to the next presentation. It's Nene et al. Profiling the diversity of the village chicken fecal microbiota using 16S RNA and shotgun sequencing data.
Good morning. I'm specifically grateful for the opportunity to come present our preliminary results to the SASAS Congress. Basically, I am Xolisinene, so we are basically profiling the diversity of the village chicken fecal microbiota using 16 amplicon and metagenomic sequencing. This is basically our outline, so you could have a good look at it. Now, for the background, chickens have lived in rural villages throughout Africa and as well as the Pacific for centuries, and have basically been adapted to the local environments and conditions. Now, these village chickens are referred to as the indigenous, native, local, or traditional poultry, and are easy to distinguish from the standardized commercial breeds. Now, location-specific, pure and nondescript indigenous chicken stocks are the first preference for rural chicken farming or rearing. Now, the chicken microbiome consists of complex and diverse communities of microbiota that influence metabolism, production, and as well as animal health. And the advancements in the next generation sequencing, NGS technologies, and the application of metagenomics has greatly enhanced the understanding of the human, plant, and animal species microbiome. By classifying the previously uncultivable microbiota and assessing compositional profiles, microbial population dynamics and microbiota diversity in the cecum. Next generation sequencing-based metagenomics resolves the limitation of conventional culture-dependent approaches. Now, the research focus has been based on human microbiome and its relationship with health and disease, while animal studies are still limited to established single breeds of beef cattle with associations to health in dairy cows, pigs, poultry, sheep under controlled environments. Now, controlled animal experiments correlating production factors contributes to variations, and the microbiome may not entirely reflect the dynamics of the natural communities. Now, the investigation under traditional management systems will enhance efforts to improve the genetic merit of village livestock, including indigenous village chickens. The aim of this study was to investigate the diversity and the composition of the fecal microbiome in village-based chicken fecal samples from Limpopo and KwaZulu-Natal using the 16S RNA gene sequencing. Now, a total of 45 16S RNA samples from 5 villages from Limpopo and KwaZulu-Natal were sampled. We used a non-invasive sampling technique so that no chickens were harmed. The selection of the chickens and the homesteads were basically randomly selected and were based on different agroecological zones and environmental dynamics. The fecal samples were collected with a spatula from freshly voided fecal samples and stored in sealable plastic bags, and then were transported to the Agricultural Research Council biotechnology platform for sequencing. And then the total DNA was extracted using the nucleospin soil kit, and then all the extracted DNA samples were quantified using qubit dsDNA kit and nanodrop for sufficient quantity and quality of input DNA for 16S sequencing. 16S sequencing was performed on an Illumina sequencing platform using the 2x150 base pairs per end protocol available at the Agricultural Research Council biotechnology platform at core. Now, after the sequencing was done, we basically received raw reads, where we then we had an in-house R pipeline for analysis, which basically used packages like Data2 as well as PhyloSeq. So for Data2, we used it for quality inspection, trim and filtering, error estimations, and for merging reads. And then for chimera checking and removal. And then for the assignment of taxonomy and phylogeny, we used the PhyloSeq database, the PhyloSeq package. And then, or basically the Silver reference database. And then for diversity analysis and visualization, we use the PhyloSeq package. These are our 16S results. Basically, if you look at it, the graph this side, which is basically the graph from this is the first poll is from KwaZulu-Natal, and then the second one is from Limpopo. Basically, if you look at the colors, they look the same, but we are going to look at them using the table so that we can then see what the basic sample abundance is in detail. If you look at it, we basically have a lot of proteobacteria, which is specified by this purple blue color, and then we also have abundances of we also have abundances of actinobacteria, which is depicted by that orange like color, and then we also have Deinococcata bacteria, basically for both provinces. And then if we move to the other side there, we have our districts. Those are your eThekwini district, your Mkambatini district from Durban, from Durban, and then we have your Mole Mole district, and you have your Ellis Mutoledi from Limpopo, and then we also have the Fitahomo districts from Limpopo. But now you can also see there's a variation in basically in the abundances. The three highest towers, they are from Limpopo. So Limpopo is showing very high sample abundances when compared to KwaZulu-Natal. These are our villages. They are showing the very high variation. There's even high variations within Limpopo province. So this is our provincial data. Basically, we have high abundances of proteobacteria. Basically, the genus is Escherichia shingella. That's the highest abundance we have, and it's from the KwaZulu-Natal. And then the other highest abundance is the proteobacteria, basically the Pseudomonas, which is in Limpopo. And the third highest abundance is the Bacteroid daughter, which are the Fermentimonas. And then at village level, the highest sample abundances we have are the Deinococcota, which belongs to the Trupera genus, and then we have the one from Umbumbulu village, which is Escherichia shingella, and then the third highest is from the Podu village in Limpopo, which basically is the Bacteroid daughter, which belongs to the Fermentimonas genus. So for the review of results, the Pseudomonas are basically the gram-negative bacteria which belongs to proteobacteria. They are basically known for their metabolic versatility and ability to colonize a wide range of ecological niches, including water environments, as also animal hosts. As well, the Pseudomonas are also associated with soil, the type of soil. So, and they're also associated with humid environments. Limpopo province is known for high rainfall as well as high temperatures, which signify the abundances of Pseudomonas species. The Escherichia shingella genus basically belongs to the phylum of Proteobacteria and are likely are the second most abundant microbial species within the provincial level. Now, the Escherichia shingella genus is there's basically two different we have two different type of genuses there. The Escherichia is basically E. coli. So E. coli, it's coli bacillosis is basically an infectious disease that is characterized by acute fatal septicemia and peritonitis affecting chickens, and is also prevalent to humans through foodborne disease transmission. Now, the Shingella species causes bacillary dysentery, which is diarrhea and fever, basically affecting the chickens. Now, the Escherichia coli and Shingella prevalence is largely impacted by rainfall, and KwaZulu-Natal is also associated with rainfall. But we should also note that the Escherichia coli and Shingella species are also they've also been identified in Limpopo, which is a province that is also characterized by high rainfall. Now, we also discovered the Trupera radiovictrix genus, which is the type of species of genus Trupera within the phylum Deinococcus. The Trupera radiovictrix grows under multiple extreme conditions in alkaline, saline, and high temperature habitats like Limpopo, which signifies their abundance in Nyakaling village in Limpopo. Now, for the conclusion, basically, the Trupera bacteriorota and Deinococcota is due to that they are normally involved in normal intestinal homeostasis, metabolism, and immunity. That explains why they they are abundant all over in between, like both provinces, villages, and in between the districts. Now, the abundances of E. coli are also present within the microbiota, which is really worrying. Now, temperature and rainfall and humidity have the effects on basically gut microbiota, because we've been able to identify the variation with the changes in temperature between these provinces. Now, indigenous chickens are a vital genetic resource and can play a worldwide role in food security. Now, it's really important that we pay attention to these indigenous chickens, because they harbor very strong genetic merits, which could prove to be very beneficial in the near future. Because I was listening to Prof. Dr. Maiwashi yesterday. He said, basically said that we'll have a very great problem of food security by the year 2050. So we need to start looking at these indigenous livestock, so that we can have the large enough animal protein to supply the population. Thank you very much.
Thank you. I think you should revisit your explanation of high rainfall in Limpopo. Most part of Limpopo has a rainfall of less than 500 millimeters. That's not a high rainfall area. So your argument that it's caused by high rainfall is not valid. You should find another reason.
That's it. Thank you. Thank you.
Thanks.
Hi, thanks. I think I agree with Prof. Scholes. I think revisit your discussion or your conclusions from your data. The temperature and humidity might affect their feeding behavior, and that might influence gut microflora. Not temperature, not humidity, not rainfall. It's a consequence thereof that might affect it. Okay, so just in future.
Thank you. Thank you.
Thank you very much for your presentation, Mr. Nene. One quick question for you. What is it unique about these Limpopo chickens against the KwaZulu-Natal one?
Well,
thank you.
Can I go on? Okay, what's really unique about these chickens is that they are adapted to a wide range of harsh, different. Okay, basically these agroecological zones, they are not the same. They are not the same. As Prof. Scholes just suggested that there's actually less rainfall in Limpopo than in KwaZulu-Natal. So that means they are both they are adapted to different scenarios, but like they are the nondescript indigenous chickens. They almost belong to the same ecotype. Yeah, I hope I answered your question.
Very well. Let's give him another round of applause. Our next presentation is coming from Dr. Ophir. The title is The Effect of Incomplete Reproduction Data on the Prediction of Genomic Estimated Breeding Values.
Thank you, Chair. As you've indicated, the title of my presentation is The Effect of Incomplete Reproduction Data on the Prediction of Genomic Estimated Breeding Values. And I want also to thank my two co-authors, Bobby van der Westhuizen and Jason Redding. It's a known fact that incomplete phenotypic and pedigree data will reduce the accuracy of our blood models that we use to estimate breeding values. That will lead to a bias in these breeding values and subsequently have a negative impact on the gain that you get within your population. And at the end, it will decrease the profitability of the enterprise and make all the effort that you put into your selections and the time you put in it worthless. For small stock, reproduction is the most important trait that should be considered during selection of both replacement rams and ewes. And the accuracy of these selection are challenging because of the fact that these traits are lowly heritable, it's sex limited, and it's only expressed after the selection was performed. And selection can further then be hampered if incomplete or inaccurate data is used for the prediction of your breeding values. The difference between reproduction traits and production traits with regard to incomplete data is very important to understand. For production traits like body weight or fiber diameter or growth traits, zero value is just a missing value. In other words, that animal doesn't have a phenotypic value for that trait. But for reproduction data, a zero value is a value. You can't ignore it because that value indicates an animal that did not lamb or did not wean a lamb. And it's valuable information that you need to include within your analysis that will have influence on the variation of these traits as well as the completeness of your data set. Therefore, we can determine that the completeness of reproduction data should always be defined as information in regard to ewes that lambed, ewes that wean a lamb, and ewes that was mated that did not lamb. All that information is therefore important, and if you leave out some of this information, it can have a detrimental effect on your analysis. The aim of this study was therefore to determine the effect of incomplete reproduction data on a prediction of genomically enhanced breeding values in the South African merino stud. The hypothesis that we worked from was that the inclusion of genotypes for a proportion of ewes will have a positive effect on the selection of replacement animals despite incomplete reproduction data. The data that was used was collected on a Grootfontein merino stud from 1968 to 2017. The Grootfontein stud is kept at Grootfontein Agricultural Development Institute near Middelburg in the Eastern Cape, and is run mainly on natural veld conditions with supplementation at strategic times. We had the ethical approval from the Grootfontein Ethics Committee. For the purpose of this study, we only going to look at total weight of lamb weaned, which is the composite trait that includes all your reproduction information. And we used the complete data set, and from that we derived 3 subsets. The first one included all the mating, lambing, and weaning information, in other words, the complete data set that was available for analysis. With the second set, we only included lambing and weaning data. In other words, and for the third set, we only used weaning data. In other words, for the last set, ewes that did not lamb, or ewes that lamb that did not wean, data was also excluded for the last one. That is sometimes the data that is being sent into for performance testing. Proc means and GLM and core procedures of SAS was used to determine the descriptive statistics as well as the significance levels of the non-genetic factors, and in the end to calculate the Spearman rank correlation between the ranks of the EBVs for total weight of lamb weaned for the different subsets. Traditional estimated breeding values as well as the genomically enhanced breeding values were estimated using the mixed 99 procedure, and we included 372 genotypes within the analysis. Here's the pointer. If you look at this data, I'm giving you all the information with regard to all the traits, but I'm basically only going to touch on total weight of lamb weaned that you looked at. If you can see there, for the total, it seems like the battery is gone. But if you look at the mean for total weight of lamb weaned for a complete data set, you can see it's 24.5. If we exclude information from ewes that did not lamb, it's come down, going up to 31.3 kilograms. And if you only took into consideration ewes that did wean a lamb, the average is almost 37 kilograms. So you can see the fact that the zeros is being moved out of the data set, they have a different influence on the mean. But the biggest problem was the effect on the CV, which was almost 58% for a complete data set, down to about 42% for the second subset, and for the most incomplete data set, only 25%. You can see that it have a huge influence on the information available and the variation within that trait. If we look at the breeding values, I'm only going to discuss the EBV for the traditional, the traditional EBV as well as the GEBVs. You can see that the range for EBVs for the traditional values range from minus 5.1 to 7.2, and for the traditional values, as the subset information decrease, the band of EBVs get narrower, with the narrowest breeding values at the most incomplete set of 4.91 versus 6.28. The same was observed for the GEBVs, where the widest range of EBVs was for the complete set of data, and as data decreased, incompleteness increased, the values, the range decreased, would get narrower for EBVs. That is quite troublesome. That means that the poorer animals, their EBVs get better, and your better animals' EBVs get poorer. So that difference between the poorer and the better animals get less. If you look at the accuracies for between the analysis, the biggest enhancement for including the GEBVs or the genomic values is the fact that the accuracies of the animals with genomics as well as the animals closer to them, the accuracies for reproduction increased dramatically. That is important because that's one of the problems with estimated breeding values for reproduction the traditional way, is the fact that the EBVs are quite lowly heritable. Quite low accuracy. However, if we look at this is the big problem. We thought that the inclusion of genomics would have an influence or limit the influence of the lack of data. You can see that between the traditional EBV and the GBV with the spearmint rank correlation was almost 96% for a complete data set. That means there's basically the best animal will be the best animal within your estimation. But if the moment we decrease the information in the within your data set, it goes down to 67% and 56%, meaning that the fact that that data is not there will not help even if you have got genotypes at the end of the day. So in conclusion, it is evident from the results of this study that incomplete data will have a huge influence on your mean of your trait as well as the variation available for selection in that trait. And despite including the genotypes of certain animals, incomplete data for reproduction will still have a huge influence on the estimation of your breeding values. It was therefore recommended for the breeds that the mating and lambing list should be mandatory for inclusion in the small stock performance testing of South Africa, and that's already being done by two of the bigger breeds, that the breeders need to include this information within the with the data set. So phenotype is king. Thank you.
Are there any questions for Prof?
So the trait that you're calling reproduction, total weight of lambs weaned, also includes growth in it. Make any attempt to disentangle the growth piece from the numbers piece?
Not for this study, no. We just plainly look at the idea for this come from when there was discussions about using reproduction data of one of the breeds for a certain study, and I just thought, I know it happened that one of the years that I submitted data, I didn't submit my mating list, and I just had the idea what happens if that information is not there. I thought it would have an influence, but not this large an influence at the end of the day. Yeah. So we didn't look at to untangle the between the growth and the reproduction is just purely to see what happened with incomplete reproduction data versus complete data?
I would just say it's just incomplete data. I wouldn't say it's reproduction per se, because you don't know why somebody didn't turn in the record. I mean, you have bad performing lambs, they don't turn in the record.
Yeah, that's normally what happens in the small stock system. The data is only loaded after weaning.
Mhm.
So most of the reproduction data is generated from data submitted for weaning, and breeders normally not know that they need to submit ewes that did not lamb or data from lambs that were born but did not wean. So that's where this discrepancy comes from.
I'd be interested to know the genetic analysis of a trait like the age at which a ewe would wean 5 lambs, would have produced 5 lambs. Her age.
Yeah.
Any other question? I'm just happy with your recommendation. They do say in the genomic selection era, phenotype is king. Thank you so much. Let's give him a round of applause. The next presentation from Prof. Michiel Scholz. The title is The Development of Biological Index for Cow-Calf Efficiency for Beef Cattle.
Thank you very much. I must first figure out which one goes forward. Oh, the green button. Good morning, ladies and gentlemen. Thank you for the opportunity. I just want to say I'm still happy to be associated with the ARC, and I want to acknowledge PLAS Media for sponsoring the shirt. Thank you very much. It is unfortunate that Johan Zitzman is not attending this, because I think this would have given him a few different perspectives. The title of my presentation is The Development of a Biological Index of Cow-Calf Efficiency for Beef Cattle, and any questions on this will be handled by Dr. Mike McNeil. Thank you. We must remember that livestock is the largest user of land in South Africa, and John Rist pointed that out yesterday as well, because more than 70% of the country can only be used by livestock. And if we want to improve food security and sustainability, we need to improve production efficiency, and that will also reduce the greenhouse gases or the carbon footprint. So it's important to improve the production per unit of beef over the whole production cycle, and not just the production of an individual animal or the growth of over a certain period. As Johan Zitzman criticized yesterday, we should not select for growth over a certain period. We should look at the improvement of the production over the total cycle. And the problem we have is to measure cow-calf efficiency in an extensive system is very difficult, because you can't measure intake. So we should find another way to do it. Under extensive conditions, an efficient cow must have low nutrient requirements for maintenance. It must be able to cope with what is available, but it should also annually produce a calf, because that's the output. So you need an input-output relationship, and that is why for many years the ratio of calf weaning weight divided by cow weight was used as an indication of efficiency, but it always favors the smaller cow. Whatever you do, if you work with a ratio, it tends to favor the smaller animal. So our aim was to develop an index for cow-calf efficiency that used weaning weight, that is the output, the cow weight, that is the input, because that tells you how much feed. The size of the cow will tell you how much it eats, and also the intercalving period. And if you have cow weight, you can translate it to large stock unit, and if you have large stock unit, you can work it out kilogram per hectare. So at the end, you can look at the calf produced per hectare if you take the intercalving period into account as well. So we used the data of the Afrikaner breed, and we used a multiple trait model to estimate the variance and covariance components and the breeding values. And the difference from most other evaluations is that we include herd year season as random effects and not as fixed effects. And in this case, your adjusted phenotype of an animal would be the breed average plus the estimated breeding value for that particular animal, and that will give you the animal's phenotypic value that it can be compared across herds. And here is the statistics. You can see we had almost 30,000 weaning weight records from 8,500 dams. Cow weight was almost 20,000, and there you have, I see this light is only working for a short while, but there you have the numbers. So this is the descriptive statistics we used. And now I'm coming to the results. Here we have the heritabilities, the red ones on the diagonal, and then the genetic correlation. And interesting thing is, Johan Zietsman yesterday pointed out, how can a survival trait like fertility be lowly heritable? Well, here you can see that intercalving period has got the highest heritability of 0.34. I'll come back later to explain why we think that happened. But there you have it. For weaning weight direct and weaning weight maternal, it's low as expected. Cow weight is 0.33. And there you can see, and now I went the wrong way. I wanted just to show you that the correlation, genetic correlation between cow weight and weaning weight direct is 0.55. The other correlations are low. Okay, if we include herd year season as a random effect, it affects the estimates of the heritability because it allows for the recovery of the genetic variance that is present between herd year seasons. If you include herd year season as a fixed effect, you can't recover the differences that are present between different herd year seasons. And this makes the prediction of breeding values much more accurate. And I'm going to show you this. We just used 3 cows at random where it was included as a fixed effect and then as a random effect. And you can see the red, if you include the contemporary groups as a random effect, the accuracy becomes higher. So that's why this technique helps us to get more accurate values when we include it as a contemporary effect rather than as a fixed effect. Then we propose this biological efficiency index. It's 365 days times the weaning weight of the calf divided by the intercalving period, or the cow weight, or the large stock unit, or the carrying capacity per hectare. You can decide. The moment you have cow weight, you can replace it with whatever measure of carrying capacity or a hectare or whatever. And if you translate it to a linear index using estimated breeding values, becomes the estimated breeding value for weaning weight direct plus the estimated breeding value for weaning weight maternal minus 0.39 times the estimated breeding value for cow weight. And remember, a bigger cow will have a higher cow weight, so that has a negative effect. But however, if you go to the estimated breeding value for intercalving period, minus 0.39, a shorter intercalving period will be minus, so it's a minus plus a minus, which becomes a positive again. And this index, we think, can work very well to estimate cow-calf efficiency. So, just a little bit of discussion. If we use multi-trait genetic evaluations to estimate breeding values for the traits such as weaning weight, cow weight, or intercalving period, simultaneously, we will get more accurate estimated breeding values. And these more accurate estimated breeding values can be obtained by including herd year season as a random effect and not as a fixed effect. It may make the model more complicated, more difficult to run, but it's better to do it that way. Furthermore, we recommend that the ratio of calf weight by cow weight or lamb weight by ewe weight be replaced with an index that includes reproductive performance. We can't ignore reproductive performance. We must include it in any efficiency index. And the proposed index that I showed you now, with the normal ratio index, ranks animals quite differently. The rank correlation is only 0.53. I think that's what you also showed. So this is not a very good rank correlation. So in conclusion, if we include reproduction in the index of biological efficiency, it makes much more sense because then we look at the whole picture. It is also recommended that the index can be derived from the genetic evaluation and its component traits. And if we use estimated breeding values to calculate cow-calf efficiency, it allows an estimate to be obtained for every individual, including bulls and young animals with incomplete records. So we can now have a cow efficiency index for bulls or a cow efficiency index for a heifer that has never produced a calf. And I would just want to acknowledge Red Meat Research and Development for partly funding this study. Thank you very much.
Any questions for Mike? You have one comment.
So this index is also incomplete. The next step in this index is to also include how long the cow lives. So the cow that has genetic potential to live for 10 years might be less efficient than the cow that can live for 18 years.
That's a very good one.
Yes.
But just the one point of clarification. So when you fit the contemporary group as a random, you improve the predictability of your EBVs, but it appears you are switching the heritabilities for production as opposed to fertility traits. The fertility, the heritabilities were like 30% for ICP, as he mentioned, but it appeared to be much lower for the winning weight.
In those data, the heritability for all the traits actually increases by including the random effect. However, depending on how much genetic variation exists between contemporary groups, the heritability might go down because the herd year season variance is in the denominator of that ratio that's heritability. So if that variance is large and there's not much genetic variance between the groups, that is, it's all environmental, then the heritability is going to go the other direction. It's going to go down. There's no guarantee what the effect is on heritability.
Good.
The important thing is you get more accurate estimates if you include it as a random effect.
Which is what you want. Okay.
You get more accurate estimates if there is genetic variance between the groups.
Okay, of course, yes.
So if there's no genetic variance between the contemporary groups, you don't get more accurate estimates.
Very clear. Thank you, Prof. Scott. It appears there are no more questions.
Thank you.
The next presentation is coming from FISA, and the title is Population Structure and Genetic Diversity Within and Amongst 3 South African Dairy Cattle Traits. And I believe you are presenting on her behalf.
Yes, I am.
Over to you.
Good day, fellow animal science colleagues and attendees of the 53rd Annual SASAS Congress. I am Chantal Krokamp, the presenting and corresponding author of this short paper entitled Population Structure and Genetic Diversity Within and Amongst 3 South African Dairy Cattle Breeds, authored by Professor Karina Visser, Dr. Simon Lashmar, and Professor Estie van Marlakoster. The South African dairy cattle industry predominantly utilizes the Ayrshire, Holstein, and Jersey breeds for commercial dairy production. These breeds have been applying estimated breeding values and have been part of multiple across-country evaluations for quite some time. The use of reproductive technologies such as artificial insemination has allowed for the use of imported semen from high-impact dairy bulls. Both government and industry investments in projects such as the Dairy Genomics Project, or DGP, have allowed for the genotyping of animals to establish reference populations for these breeds. Genotypes available for these breeds include those of both local and international bulls. The sharing of genetic resources between high-impact dairy bulls and their progeny affects the genetic diversity amongst populations. It is therefore important to investigate the current genetic architecture and existing genetic diversity, or a lack thereof, for future selection strategies and to ensure the conservation of genetic resources and viability of future South African dairy herds. The aim of this study is to perform a SNP-based genomic analysis of 3 South African dairy cattle breeds, namely the Ayrshire, Holstein, and Jersey, using population structure and diversity parameters. SNP data was available for these breeds, which consisted predominantly of South African animals with a few international animals. As indicated in Table 1, the number of animals included were 535 for the Ayrshire, 1,257 for the Holstein, and 1,133 for the Jersey. The Illumina bovine SNP 50 version 3 B-chip array was used to genotype the animals participating in the Dairy Genomics Project and subsequently those used in this study. A set of 51,448 autosomal SNPs were retained for downstream genomic characterization. After standard quality control in PLINK, observed and expected heterozygosities, as well as mean minor allele frequencies and linkage disequilibrium, were calculated. Principal component analysis for genetic relatedness was performed with genome-wide complex trait analysis, or GCTA software, and admixture was used for ancestry estimation. Table 3 indicates the different parameters and respective threshold values used to obtain the relative results in PLINK and admixture software. Individuals were removed if MAF values exceeded 5%, and both sample and SNP call rates were less than 95%. LD pruning was performed after initial LD analysis, but before PCA and admixture analyses. The LD prune data considered all pairwise comparisons with no lower limit regarding the R-squared value. However, those exceeding an R-squared value of 0.5 were removed due to too much linkage. The observed heterozygosity ranged from 0.336 in the Jersey to 0.364 in the Holstein breeds. For the Ayrshire and Jersey breeds, the observed heterozygosity was 0.02 and 0.025 lower than expected heterozygosity, whereas in the Holstein breed, they showed a negligibly small gain in genetic diversity of 0.003. The mean MAF values for the Ayrshire, Holstein, and Jersey breeds were 0.22, 0.23, and 0.19 respectively. The longer-ranging R-squared values of LD, so those ranging the longer-ranging R-squared values of LD, so those smaller than or equal to 1 megabase pair in length, ranged from 0.123 in the Holstein to 0.161 in the Jersey, whilst those of a shorter range, so those smaller than or equal to 100 kilobase pairs in length, ranged from 0.236 in the Holstein to 0.281 in the Jersey. So the PCA results, as shown in figure 1 over here, illustrates 3 separate clusters, with most outliers being predominantly in the Holstein breed. This is supported by the population admixture estimates, as shown in figure 2, where 3 separate ancestral populations, represented by K is equal to 3, are visualized in red for the Ayrshire, green for the Holstein, and blue for the Jersey. The overall results indicate that the greatest genome-level diversity is found within the Holstein breed, followed by that of the Ayrshire and then the Jerseys. This is supported by the inclusion of a greater number of international animals in the sampled Holstein reference population used in the study, as compared to that of the Ayrshire and the Jersey. Weaker LD levels for the Holstein are indicative of smaller haplotypes shared amongst South African and international animals, possibly due to younger and less influential animals and a lack of persistence, long-range LD between countries, or in other words, the genomic diversity between global populations. The outliers in the PCA results could further support less genetic relatedness between local and international animals. Similarly, the tighter clustering observed in the Ayrshire and Jersey breeds can be attributed to fewer international animals within these respective reference populations. In conclusion, the analyzed results support the influences of globalized dairy genetics on the diversity of the Jersey, Ayrshire, and Holstein reference populations in South Africa thus far. Since the erosion of genetic diversity limits long-term genetic gain and impedes sustainability amidst future challenges, these results may assist in strategies to improve upon and update reference populations for genomic selection. Thank you.
Thank you for the presentation. Are there any questions? Mike.
So do you actually know that those outlier dots in the PCA are the international animals?
Yes. So from the data that we received when we did our initial analysis, we went and looked at that, and those are bulls from countries such as Germany, Russia, Canada, America, New Zealand. So there's a much wider range of international bulls included in the Holstein
Okay,
population.
so those dots
yes.
are actually the international animals.
Yes.
So that leads me then to, given the fairly high degree of willingness to share genotypes internationally, why did you not sample the Jersey, Ayrshire, and Holstein from, I don't care where, Ireland, the US, Germany, and have groups of those animals represented in your diversity analyses?
So this is just a preliminary phase of this whole study. Professor Visser and Dr. Lashmar and Professor van Marlekoster are still currently working on this. This is just what we have presented thus far for the SASAS Congress. So the study will still expand further. So for now, we just had the South African data, so that's what we analyzed.
Okay. Thank you for the wonderful presentation. I just wanted to know why do you think the Holstein breed had the highest genetic diversity than the Jersey?
So obviously we wouldn't expect that given in, the Holstein breed in general. But we speculate that it has a higher level of diversity due to these international animals and less relatedness between them because they stem from different countries instead of just South Africa, if that makes sense.
Maybe just to help on Mike's question, follow-up question, that attempt was made to sample from breeding company, or rather to request genotypes or samples from those international companies, and that was not, there was no willingness to provide that sample.
Okay, thank you very much for your presentation. Just a quick one. When you performed this analysis, did you consider to select for unrelated animals when you perform this genetic diversity study?
That is actually a good question, and I can get back to our main author regarding that question because Dr. Simon Lashmore actually did the analysis on this. So what we received is basically we just asked for genotypes, and genotypes were selected from the reference populations and sent to us by the company. So we didn't have a role to play in which animals are related or not. But generally there is a relatedness at some level or other. So yeah, thank you so much.
Thank you. Let's give her another round of applause. There is one announcement. There will be an award ceremony after this session, so we are requested to remain seated for the poster awards. The next presentation we'll hear now from Nell and the benefit of marker data in the prediction of production traits in South African merino, SSG blab versus A blab.
Green button. Good morning, everyone. My name is Cornelius Nell, and thank you, Chair, and thank you very much to all of you for powering through again to the Wednesday session, which is very often overlooked as people start to head home early. I know we're pushed for time, but I think it's appropriate to quickly note that this whole presentation only includes one slide of results, which does not look like much. But scraping together the resources to allow for studies like this has been the product of a lot of effort from various individuals over many years, starting even before I joined the program, including individuals, but not limited to the long list of co-authors listed here. So the title of the study has been read by the chair, and we'll be looking at the benefit of including marker data in the prediction of production traits in South African merino sheep. Now, genomic selection capitalizes on the ability to capture genetic variation at a molecular level by additionally incorporating marker data into our genetic prediction models. Now, they deliver new opportunities to enhance the ability to predict candidates for genetic selection, perhaps in ways that traditional blood methods have proven to be limited. However, sorry, that's really distracting. Sorry. Excuse me. New is not really new anymore. Genomic selection has now become commonplace in a really wide variety of livestock species worldwide, and this has been the case for many years now, and including for sheep. And as an example, the use of marker data is really well established in the national genetic evaluations for the most important sheep breeds both in Australia and New Zealand. But when we're evaluating the prospects of genomic selection or the use of marker data, it's very important that we don't superimpose results from other species, which I see is commonly the case. When people sell genomics, they tell you with the success they had in species like dairy or pigs, and which is, I think, not so really appropriate. Amongst the determinants of the accuracy you get from genomic selection is, for example, the genetic diversity or the effective population size of the species that you're working with, and pooling sheep as being in any way similar to these breeds is really not, to me, a sensible approach. And this is particularly the case of a breed of very high genetic diversity, as the merino has been shown to be. So, yes, it is useful to look at examples elsewhere, such as Australia, when the progress they've made with genetic selection and genomic selection for merinos there. But of course, we would want validation on a local basis because each situation is unique. But in South Africa, the current scenario is that the research sector, has been really been responsible for the only dedicated genotyping strategy of sheep and mostly merinos in South Africa. Now, from the big picture, this might not be entirely ideal, but we believe that the research sector could still really be beneficial to the development of genomic selection methods in South Africa. First of all, just by proof of concept, which is mostly how the research sector and our resource flocks have operated over the years. But second of all, perhaps by directly participating in the development of genomic selection methods in South Africa, by directly contributing resources to the establishment of a reference population that we can use for reliable prediction of traits in South African merinos and eventually perhaps other sheep breeds. So there will be multiple challenges associated with this. These resource flocks are managed as selection experiments, so often they are isolated and their structures are different, but which is just one of the possible problems. But that, and also the concatenation of data from different resources, such as different resource flocks, and different sectors, such as both the commercial and the research sector, has not really been tested. There could, however, also be multiple benefits to this approach. So we could develop genetic selection or improve genetic selection methods with the goal of improving our prediction of production, or the so-called economically important traits. But we could have a dual objective running in parallel of eventually expanding our goals of selection to include difficult to measure traits, which up until now is also something that's been restricted to recording only in resource flocks, and perhaps we can make use of this information to the benefit of the wider sector. So the objective of this study was to deliver the first validation of the use of genomic information in the genetic prediction of SA merino sheep. We will do this by combining data from multiple sources, including different flocks from the resource sector and different sectors, being both commercial and research data sets. Our target for the moment is limited to production traits, and this is perhaps not the ultimate goal of genetic selection, because you would be forgiven of making the argument that traits that have a moderate to high heritability are already easy enough to select for. So perhaps the very expensive investment in genomic into producing genomic data is not worth it. But for now, when we're testing the method, I think it's reasonable to say that using traits that are well established, that we know what to expect from them, is useful when we are in fact comparing the method and not evaluating the traits themselves so much. So our sample populations were split up into two groups. We had 5 resource flocks, known as the Elsenburg, Grootfontein, Kraddock, and Tygerhoek experimental flocks, and the Langgewens flock, which is perhaps more a population rather than a stud. And then we also had 5 commercial flocks that currently contribute data to the National Small Stock Improvement Scheme. We did select these flocks as an overall representation of South African merinos, but it's important to note that they were also selected by an additional criteria that they do have some linkage to some of our resource flocks, which has been something that's been in development for quite a number of years. Them they've just labeled industry 1 to 5. We won't name them, but the flock names are also not going to be important in the results as we present them here. Phenotypic data, as I said, it includes only production traits, but this leaned up to a maximum of about just over 70,000 records for weaning weight. Then we also included another weight trait. We had just yearling weight, and then 3 wool traits, fiber diameter, clean fleece weight, and staple length. So the genotype data was also a long process, which has been happening for across a long number of years, and consequently we have used different platforms for genotyping in terms of chip choice. And this has indeed left us with quite a messy imputation program over the years. And we started on the Illumina ovine SNP50 chip, and eventually ended on the GeneSeq genomic profiler chip, and imputed across chip platforms eventually to settle on the GGP chip, which was our choice, since it's currently also the cheapest option, and also to be more in line with our colleagues in Australia, which also made the switch from the Illumina chip. After the standard quality control procedures, we ended up with about just over 40,000 SNP markers for 2,800 animals, of which 277 were sires and about 2,600 were dams. About 1,600 records came from resource flocks and about 1,200 from the industry flocks. So the ultimate aim was just to compare the prediction accuracy of our traditional methods, which we'll call A-BLUP here to distinguish from single-step genomic BLUP methods, which we'll call SSG BLUP, as it's commonly referred to. The A-BLUP works just with the standard numerator relationship matrix. We use a full pedigree of about 88,000 animals. In SSG BLUP, we combine this data set from the phenotypic and the pedigree and the genomic information into one in the very standard method of evaluating the H matrix. I show the equation here only to show that this alpha parameter was scaled to about 0.95, which means we really quite heavily weigh genomic information in the definition of H inverse. Also, and as a note, G, of course, was just scaled so that the means of diagonal and off diagonal aligned with A. Also, I can't go into all the particular analysis. We don't have the time, but generally speaking, we're looking at standard linear mixed models. And the only difference, so we would test fixed effects and then test random effects, and then eventually the only difference between the two analyses would be replacing either A with H. And the two methods were compared by cross-validation methods, which is also quite common. And what we're eventually trying to do here is to replicate the selection of unproven candidates who have no records by assigning a validation population somewhere in our data set and removing these records and setting them to missing. We do one single whole analysis with all of our records when we get our vector of solutions, which we call AW, which is the whole breeding value, or we'll, for the purposes of this analysis, call the true breeding value. And then after assigning a validation population and setting the records to missing, we repeat the analysis exactly as is, and for the same validation animals, we derive a part breeding value, because that's the partial data set. And accuracy here was determined according to the LR method, known as the linear regression method, also neatly lines up with the surnames of the authors, known as the Legara reverter method by many. Similar to the correlation, but scaled differently, using the correlation but scaled differently a bit in the denominator. So we had two validation designs referred to here simply as scenario 1 and scenario 2. In scenario 1, we looked at a forward validation, which is also quite common, where we just keep all the records from all the flocks, but by year we choose a cutoff date, and all animals born after the cutoff date are assigned into the validation population, and they'll eventually, for the part analysis, have their records set to missing. It's difficult to align it perfectly, but we generally aim for having about a 25% partition of phenotypes and genotypes set in the validation population, and in that case, we had it set at 2016. Animals born 2014 or later assigned into the validation set, which ended up with about 16,000 weaning weight records, if we're using that as an example, and about 600, of which about 600, just over 600, was linked to genotypes. So in scenario 2, we looked at something else, which we call the cross-flock validation. This is quite similar to K-fold validation methods, unless there's no random clustering or random assigning or clustering methods involved here. You're simply assigned into the validation population if you were born in a particular flock. And then we repeated this analysis, this part analysis for each flock. So then 9 flocks went 9 times the analysis, and eventually all animals had a part and a whole breeding value according to this design as well. I say 9 now, which is not 10, because the Langgewens flock was excluded from being considered a true flock in this design. And here, of course, the amount of records in the validation population ranged quite considerably, but relatively speaking to scenario 1 was quite small. So here we can see all of our results summed into one bar plot, where your traits are aligned on the x-axis and the accuracy of prediction aligned on the y. The methods are aligned side by side with SSG BLUP in the dark blue. Clear to see, first of all, of course, that accuracy of prediction varied widely across different traits, which is expected given different heritabilities. But for scenario 1, which we'll look at first, SSG BLUP outperformed A BLUP by useful margins for all of our traits, but by highly variable margins depending on the traits that we're looking at, with really rather distinct differences, for example, between fiber diameter and weaning weight. Also, the margin of gain here aligns slightly. Previous studies would often report that the traits with the lowest heritability has the highest to gain from genomic selection. More or less true here, roughly, but the traits did not line up according to this ranking perfectly. Then for scenario 2, it was clear that not having within flock information had some severe implications for overall accuracy across all of our traits for both methods. But, however, this is where we saw larger proportional gains from having G-BLUP or SSG-BLUP methods, and a particular example being weaning weight, where the accuracy of selection actually doubled. This is a good result, but I think it's important also to keep in mind that proportions become a funny thing when we're working with small values. But even if we're just looking at the absolute values, I think an interesting result here was that for most traits, I think except fiber diameter, the SSG BLUP result from scenario 2 was able to at least match or exceed the A BLUP result from scenario 1. So with the help of genomic methods, we were able to obtain a selection accuracy similar to the traditional within flock selection for most of these traits, which I thought was quite a positive result. So this study provided the basis for multiple new developments. It was the first appropriate validation of the use of SNP data in genetic selection for South African merinos and I think in sheep in general. It's also the first cross-flock analysis of this kind for South African resource flocks, as well as the first combination of data sets from the industry and resource sectors. In scenario 1, the use of marker data did benefit the accuracy of genetic prediction, and despite a relatively modest reference population size, the amount of genotypes compared to international papers, we thought that the gains made by SSG methods were still very useful. But a note of caution is worthwhile, since we're working with population-wide metrics, and that's useful when for testing methods and then applying to genetic gain. But these results are a product of the structures that we're working with right here, and there are an infinite amount of moving parts that could change if we were to repeat the same thing with different animals, different populations, and even a different experimental design. These results are subject to change. So I could also note it's not safe to look at, but results on a per flock basis were also quite variable, and this exact amount of increase in accuracy should not be assumed universally. And as such, I think continuous validation like this should be a key part of development of genomic methods in South Africa, for sheep especially. So scenario 2 is probably not the most real-world scenario currently, and in a way probably a little bit conceptual, but important because it tells us how can we translate information usefully to across the cross-flock boundary. Which is important in sheep, where flocks can very often be genetic groups. But it also is of particular relevance if we're aiming towards the development of selection for difficult to measure traits, because, well, they are by definition not easy to measure, and perhaps if we can predict across flocks with a reliable accuracy, we can limit the measurement of difficult to measure traits to only selected flocks within our bigger population, which would be a very useful thing to approach for the future if we're looking at a wider scope of selection objectives. It was clear that here genomic methods was a very useful tool to increase this capacity, and it aligned with the literature where genomic selection is very often capable of some moderate accuracies where ABLA proves to be very, very low or very, very limited in its capacity for prediction. But some threshold to this benefit is expected, since obviously also genomic prediction has limited penetration across very distant populations. So in combined with perhaps investment into genomic resources, it's highly recommended that this should also be joined with dedicated efforts of cross fertilization. So
you need to wrap up now.
I'm done. With better linkage between our groups, between target and reference animals, and that is the. Would just like to thank our contributors to the study, and then I'm done. Thanks.
Thank you for the presentation. Unfortunately, there's no time for questions. There's 3 minutes deficit. So we'll have to move on to the next presentation. The name is Nemotandani, Estimation of Breed Effects and Non-Hereditary Genetic Variation for Ostrich Slaughter and Skin Traits.
Good day, ladies and gentlemen. I'm Keto. I'm going to present a estimation of breed defects and non-additive genetic variation for ostrich slaughter and skin traits. I will have a short history regarding the ostrich industry. South African ostrich industry started early in 1857, with the main focus being on feathers. Later on, the interest shifted to meat and also the leather. Overall, South Africa is home to 70% of the world ostrich population, and it's a export industry with 90% of the product produced in South Africa being exported. And the main product in ostrich industry is meat, feathers, and also the leather. Presently, the global industry is facing a huge challenge when it comes to skin quality or leather quality. Most of the leathers that are produced presently are being downgraded due to the fact of defects. Defects can be scars or also markings or pitting. Crossbreeding improves the performance of hybrids over the mid parents. And the structured crossbreeding plan can improve the productivity in the ostrich industry. Previous studies have shown that heterosis is possible. And then there's 3 different breeds. On the slide they look the same. However, there is SAB, which is South African black, Zimbabwean blue, and Kenyan reds. The difference there is their sizes, with the Kenyan red being the tallest, but smaller in size to the Zimbabwean blue. And South Africa industry is dominated by the South African black. And lately, there is introduction of Zimbabwean blue and also the Kenyan red. The aim of the study is to quantify additive breed effect and the putative non-additive genetic variation for quantitative and qualitative skin traits in 3 pure breeds and also between their reciprocals. And then the pedigree and performance data was recorded at the Oudtshoorn Research Farm Ostrich Resource Flock from 1998 to 2021. This is in the Western Cape. This the records that we have. Like I said earlier on, that SA black, South African black, is the most dominating breed in the industry. You can see also on the number of the blues and also the reds. Traits evaluated, slaughter and skin traits were measured we weighed the live weight, skin size, skin thickness, and also the crown and neckline traits. Subjective traits were scored: nodule size, nodule shape, and hair follicles from 1 to 9, with 1 representing for hair follicle, with 1 representing less hair follicles and 9 excessive hair follicles. Data analysis: sex, genotype, and the interaction between the two was the effect. Slaughter age was used as a covariate. The pure breeds were pure breeds, and also the combination between South African black and Zimbabwean blue, and also combination between South African black and the Kenyan red were analyzed separately to assess the crossbreeding effect between the breeds. And then 2x2 diagonal crossbreeding were designed to assess the crossbreed combinations and also degrees of freedom for the genetic group were derived. They were used to derive linear contrast for effect of additive breed effect, heterosis, and also Demline effect. This was my descriptive statistics with farm weight. The mean there was 101, while for slaughter weight was 87. And then for crown traits, it was, let's say for skin grade, it was 3, while for for hair follicles, it was 3.6. This was the predicted means for all the purebreds, which is SAB, ZB, and also the KR. From this table, it shows that the slaughter weight, there was a significant difference for slaughter weight with ZB and KR outperforming SAB, which means they are bigger than the SAB. And then for skin thickness, skin thickness was not significant. Basically, skin thickness is more determined by the age, the sex of the ostrich influences the skin thickness. And then the crust size. Remember, like I mentioned earlier on, that the SAB is smaller as compared to ZB and KR. That's why there was a significant difference between for skin size. For crown length, there was a significant difference. Like I said, Kenyan red and Zimbabwean blue, they are bigger in size and they are taller than the SAB. And then also for the neck total length, due to their tallness and bigger size as compared to SAB. And then for subjective traits, these are the traits that you usually determine the quality and level of the skin. Because this is the reason why most of the skin traits, most of the skins are being downgraded. If the nodules are well developed, chances are you'll get good money. And then if the hair follicles are less, still it also means that you'll get good money for the skin. And then in this case, the ZB and KR for nodule size, they were much better as compared to SAB. And for hair follicles, KR was even higher score. Higher score in hair follicles is something that we don't need, because the more the occurrence or the presence of hair follicles on the skin, it affects the quality and also the grade. And then for SAB and Zimbabwean blue and their reciprocal crosses, slaughter weight was higher for cross of ZB and SAB, and also there was also heterosis, meaning that the performance of the offspring was higher than the mid parent. And then also on the skin weight, there was quite different, where the crossbreeds were performed far much better than the SAB. And then for crown length, there was effect on the additive British. There was effect on the group there with the crossbreeds performing much better than the SAB. And then also on the neckline, neckline total length, the breed effect there was visible with the crossbreed performing much better than SAB. And then for subjective traits, for hair follicle, I can say that the SAB was far much better. Then, as it is more suitable to have less hair follicles so that the skin quality is not affected or is not downgraded. However, on the nodule shape, the crossbreed, on the nodule size, the crossbreed was much better than SAB. And then for SAB and Kenyan Reds, the slaughter weight for the crossbreeds was higher compared to the SAB or to the purebreds. And then also on the skin weight, the additive breed defect observed with the crossbreeds having higher values as compared to skin weight. And then for crown length, there was a thoracis. For KR, crown length, remember, I mentioned that the KR or Kenyan Red is taller as compared to the SA Black. Right, that's why there is a thoracis observed. And then on the neck total length, the additive breed effect observed there, and then with the crossbreeds having higher values as compared to SA Black. And then on the nodule or subjective traits, the crossbreeds outperformed the SAB, and also, however, when it comes to the hair follicles, SAB was much better as compared to the crossbreeds. And then, when we include weight as a covariate, for purebreds, most of the significant levels was reduced on all the traits. And then for SAB and ZB and the reciprocals, most of the trait thoracis was insignificant. And then the hair follicles, the thoracis for hair follicle was still significant, but it was reduced. And then for SAB and Kenyan Red and the reciprocals, there was a significant reduction on a thoracis for slaughter weight, crown size, crown length, and crown shape. It's mostly for size-related traits. In conclusion, ZB and KR beds outperformed the SAB on size-related traits, and then commercial breeding could improve slaughter weight and other size-related traits. Subjective quality traits were either improved in ZB and KR or their crosses or similar to values recorded in SAB strains, except for hair follicle. Higher growth rate and feed intake, however, between genotypes still have to be investigated further. Overall, crossbreeding may thus benefit commercial ostrich industry or ostrich production in a structured system based to scientific information. I would like to acknowledge the following institution, and then thank you.
Time for half a question.
Keto, thanks. Quite insightful. Just a quick question. What, maybe I missed it, what was the age of the birds that you included in the study?
There was quite range from, I think, from 7 months up to 20-something months, I think so.
Do you think the picture will change if you only include the industry, the general industry slaughter age of birds?
Presently, it's when it comes to slaughter age, it's quite challenging because nowadays with the farmers are trying by all means to cut the cost, especially the feeding cost. Presently, they are slaughtering at 9 months, 8 to 9 months, and 8 to 9 months. By that time, the nodal, the skin traits, most of them, they have already, or they're already on an acceptable state. I think it can actually.
Just quick. Just be careful how you put it into context because if you, let's say, industry return to normal, the picture might change entirely what your results might mean. So it would be nice. I know you're going to defend your thesis soon in the department. So I would like to see a return on investment calculation, please.
Thank you.
Thank you. And thank you for the nice presentation. We don't take the tools, please. We need that. The next presentation is in Ngapeli, effective population size, inbreeding coefficient and risk status classification of Kalahari red goats from some years in stud flocks of South Africa. Okay.
Good afternoon. I'm Katlego Ngapeli, and I'm going to be sharing with you my project. I was working together with Nimtandani and Professor Tada. The effective population size, inbreeding coefficient, and the restatus classification of Kalahari red goats in stud flocks of South Africa. Goats were one of the first animals to be domesticated, and this was due to their temperament and their productivity, including their nutritious sevon, milk, wood, hides, fiber, and the leather. Hence, we have 5.62 million goats in South Africa. The most popular indigenous goat breed meats in South Africa are the Boer goat, the Kalahari red, and the Savannah goat. And the Kalahari red goat Stock Breeders Association was actually developed in 1999, and since then it's been functional. Co-ancestry actually affects the population structure in such a way that we end up with loss of genetic variation for future populations. And as a result, we would end up with low quality of breeding stock and limited supply of genetically improved animals. And this also is affected by the lack of technically trained personnel in integrated goat farming systems. Inbreeding depression affects the goats in such a way that when there's 10% inbreeding rates, the animal's productivity is actually going to be affected by 20%. And as of 2014, there were 29 extinct goats and 11 that were threatened throughout the world. So therefore, it's important to conduct the study because the genetic contribution of an animal is one that determines the maximum potential of the particular animal, and the environment also fine-tunes the genetic expression of the particular animal. Now, studies have been done to check the effective population sizes, but not on the particular breed, the Kalahari red goat. Genetic drift has also long been reported. Therefore, it's important to know the acceptable ranges of inbreeding. My objectives were to determine the inbreeding coefficient of the Kalahari red goats in different generations from 1977 to 2018 in South Africa, and to determine the effective population size of the Kalahari red goats in stud flocks of South Africa from 1977 to 2018 as well, and to also ascertain the restatus classification of the goats. The breeders of the goats were actually distributed throughout the country, and with the breed being the Kalahari red goat, you can see a typical picture of the dew and the bug. And then for the sampling, it was a total population sampling technique. Only the animals with duplius effects were removed from the population, and all the other animals within the population were actually kept. The reason being that if we're checking inbreeding, we need to check the lineages from the base population until the current population that there is. The data was obtained from the Kalahari Red Breeders Society and also from Stud Book, with 26,203 animal sets in 17 generations and 72 breeders. I gathered the animal's ID, the sire, the dam, date of birth, sex, breeding males and females. After editing, this was the structure of my data, with 16,793 females and 9,410 males. Also, the reference population was 17,945. These were the animals that had both the parents recorded. And then the ancestors contributing to the reference population was actually 2,469. To analyze the data, I used NDOG version 4.8, and the formulas indicated for the generational interval, for the inbreeding rate, inbreeding coefficient, and the effective population size. And to also to check the restatus class, I used a rubric which was provided for by DAF in 2012. My results stipulated that the average inbreeding was 4.03%. Was 4.03%, and out of the 4%, 0.36 was from full siblings mating, 3.98 percentages was from half siblings mating, and 1.21 percentages was parent to offspring matings. The average, the mean inbreeding was 2.65%, and mean relatedness at 1.80. The expected inbreeding caused by unbalancing of founders was 0.32. The maximum inbreeding coefficient, the minimum was 0% in generation 1 and 2. Reason thereof was it was only it was then that the society was being introduced. So definitely the animals that were being recorded were still new. It was like the base population. And then in the 7th generation, that's when we experienced the most inbreeding. And here is how my inbreeding coefficient was distributed. So after the 7th generation, we can actually see the population structures were dropping, and the worst out of all the generations was actually the 16th generation, where there was only 41 recorded Kalahari red goats. The information is now depicted on a graph. So yes, after the 7th population, that's when now the population structures was actually going down, and this actually affected the inbreeding levels, because after the 7th generation, we can actually see that the inbreeding rates were actually increasing. And then for the effective population size, the base population was 17,945 goats, as previously mentioned, and of which the ancestors was 2,469. The base population, one that had more, one or more unknown parents, was 8,258. And then to classify the effective population size, if the goat population is more than 1,000, we then say the breed is secure. If it's between 1,000 and 500, it's safe. And then 500 and below, it is threatened. On the graph, we can see that throughout the generations, the population structures were safe and secure, but from the 16th generation, it actually dropped beyond the acceptable range. For the race status classification throughout the 17 generations, as you can note, in the first generation, they were not at risk. The population was balanced, but then from the 16th generation, that's when it was critical. And as mentioned previously, that there were only 41 animals recorded, and of which the male to female does not balance as well, as we had more males than the females, meaning even if we wanted to reproduce, the females actually had a more limited number as compared to the males. In conclusion, the inbreeding coefficients are not the same throughout stud flocks of Kalahari red goats, and the effective population size of Kalahari reds in South Africa are not the same. The Kalahari red goat is currently endangered, meaning that should the breeding trends continue in a similar manner, we might end up losing the breed. And then therefore, the higher the effective population size, the lower the inbreeding coefficient, and the risk status of the animal will be secure. I would recommend that intense conservation of the breed is actually implemented to also increase the population, that we maintain and improve the elite breeding males and females, that advancement of statistical methodologies for genetic evaluation be done, assessment of genetic processes, genetic progress, the development of breeding objectives for the Kalahari red, and assessment of inbreeding depression in traits of economic importance. I'd like to acknowledge the indicated institutions. Thank you.
Okay, there's some time for questions.
Yeah. Thanks, Mike. Just a quick question. Why do you think the fluctuation in, let's call it endangered status of the Kalahari red?
The fluctuation.
Yeah.
There's in other years there's like less participation. I think some breeders would not be participating because for various reasons. If the breed as is not in the market, it's not selling as much. Other breeders who are actually breeding on it, who are focusing on breeding it, would actually be discouraged. So therefore, having to register and might feel tiresome for them because they might not feel like they're not benefiting as much.
Okay, so it's a data phenomenon and not a natural one. That's actually what I'm asking.
Yes.
So, in papers like this, in research like this, the incomplete records become very important.
Okay.
And in this case, incomplete records are those which the parents are unknown. So in some of the later generations where the inbreeding went down, is that due to incomplete records? Is that due to unknown parents?
I would say no. Reason being that the ones with unknown parents, they can still be used as the base population, provided it now had like offsprings in the next generation, and so now we can actually keep it as the base population. So the ones that were removed are those that had records that did not make sense. You wouldn't get when it was born. You wouldn't get, yes.
So then how did in generation, I don't know the number, 10 or 11 or whatever, how did inbreeding go down so dramatically? Why?
Why did it go down? I would say it was after having such a high inbreeding rate, right? I guess you get to see the effects of it. You can see the productivity drop because after having something like 6% and more of the inbreeding, you can actually start seeing the effects of it. And therefore, you can now be aware of the fact that when you breed for the next generation, you don't have to breed within your particular flock, but then that you can outsource genetic material from the other farmer and then like introduce in yours.
I think my question follows on from Mike's question. I'm wondering about flocks exiting and entering from previous experience using statbook data. What is the possibility that flocks exiting and entering the data again might have contributed to some of your results?
Each animal has an animal ID. So even if it exits and comes back, we still have the animal ID. So we'd still know this is the animal that was previously recorded. We'd know that's still the same animal.
That are mated in the period it wasn't in the scheme would enter the system as base animals.
Yes.
Okay. Well, good job. Let's give her another round of applause. Just a reminder, we are asked to remain in this venue for the poster award. So we are moving on to the last presentation of this session, staying to the title of the presentation is Genetic and Environmental Parameters for Birth Weight and Behavior of Neonatal Merino Lambs in relation to cold stress. Is it? Scalp, you are here.
Oh heavens, how do we work this? Okay. I'm Skal Klute and I'm presenting on behalf of Shannon Stein. I think she's prettier than me, but you have to put up with me anyway. So we know that lamb mortality is a significant welfare and production issue, constrains ethical and sustainable sheep production, but we also know that lamb survival benefits from early suckling and colostrum intake, which is impaired by inclement weather. So there is a lack of published findings on the effect of cold exposure in neonatal behavior and progress to suckling in lambs. So the aims was to use historic behavioral data to establish a relationship of neonatal lamb behavior with cold stress, and also to derive genetic parameters for lamb behavior. And our hypothesis was that cold stress impairs early suckling activity and that lamb behavior is heritable. Okay, so we used the Ellsberg flock, an experimental resource flock. Neelis has also used some of it. And it has been divergently selected for reproduction, number of lambs weaned per ewe joint since 1986. Initially, they were randomly screened from another resource flock, the secondary to primary follicle, wool follicle flock. And there were basically two selection lines, and we used Helen Newton-Turner's formula to differentiate between the progeny of ewes that had more lambs than they had lambing opportunities, and in the high line and in the low line, the selected individuals would be ewe from descendants from ewes that had less lambs than they had lambing opportunities. In other words, they either lost all progeny born or were barren at least once. And I think it's a valuable example of selection in favor of animal fitness and resilience. Now, this is just an indication of genetic trends. Now, is this the... No, this is going the wrong way. Just have to figure out where. Okay, here it is. So okay, we looked at the component traits of number of lambs weaned. So conception here, multiple birth there, and then the composite trait. You can see that okay, the responses was more or less symmetric for ewes, land per ewe joint. About 0.5% per annum for multiple birth rate, and in the downward selection line there wasn't a lot of change. In ewe rearing ability, about 0.15% going up and 0.23% going down, and then about the percent going up in the composite trait, and about 0.5% going down. So this is just to give you indication of the responses that accrued over time. Now, in this study, we used neonatal lamb traits, the standard trait birth weight, and then behavior latencies, first birth to first standing, and then standing to first suckling. Now, this was transformed to natural logs after 3 was added because they were very low values in some cases. And we used records of individual lambs within years and due cooperation with the first suckling attempts. And this was scored on a 1 to 6 scale. So we have 3 behaviors conducive to early suckling, which is standing still, adopting a slightly hunched position to give access to the udder, and then also nudging the lamb in the correct position to suckle. And then, of course, there are 3 unwanted traits. This is circling away from the lamb, backing away from the lamb, and also being aggressive towards the lamb. So the absence of the bad traits and the presence of the good traits give you a score of 6, and you had to stand still at least when the lamb suckled, so a minimum score of 1 and a maximum score of 6. Okay. So we obtained climate data from the Elsenburg weather station. There's some information, and it was basically, let's see if I can. So it was based on wind speed, temperature, and then rainfall, the index. Now, the genetic analysis, so pretty standard for the fixed effects, but cold stress was added as a fixed linear and or a random cubic spline component. And then the random effects was additive effect, maternal, and then permanent environment or the common environment provided by the U. Okay, so this is an indication of what it was, pretty standard for birth weight, and you can see, now I'm going the wrong way, and you can see a lot of standard deviation exceeding the mean for the latency traits, but that was pretty well dealt with by transforming as I indicated. We looked at this, and birth weight I'm not going to comment because that is what we expect in most cases and was based on historic line differences as well. When we look at the behavior latencies, so you can see the highline lambs took shorter to about 5 minutes shorter to progress from standing to suckling, and then multiples also took a bit longer because of competition with the other sibling, that sort of thing. And then finally, just an indication that dam cooperation resulted in a higher score, give you shorter latencies for standing to suckling. Okay, so when we look at the effect of the cold stress, only the fixed linear component was significant, and it was not in interactions with class variables. So what I did is I predicted means at relatively mild conditions of 800 kilojoule per square meter per hour, and well, let's say some of the most severe cold stress that we experienced, which was about 1200, and interestingly, the latency from birth to standing was actually reduced at higher levels of cold stress, but the latency from standing to suckling was increased quite substantially by cold stress by about 70-odd%. Now, the reasoning that the latter was expected because that is pretty well covered in the literature. What we think what could have happened is that under cold conditions, the lamb may be prompted to try to stand as quickly as possible in an attempt to try to top up dwindling resources, and that might have resulted in them, trying to suckle earlier, trying to stand earlier. When we looked at the genetic parameters, for birth weight, it was pretty standard. The interesting thing is that it was quite high, actually, for the latency from birth to standing. not a lot of genetic variation for latency from standing to suckling. At the maternal level, there was quite high maternal heritability for birth weight, lower for birth to standing, and then the common environment was the only maternal effect that was significant for standing to suckling. So this is pretty much a maternally influenced trait. And then when we looked at the correlations, not really any correlation that was significant. And so, to conclude, I think cold stress compromised the latency of standing to suckling in neonatal lambs. And this explains higher levels of lamb mortality when we are experiencing cold stress. And selection in the H line improved the ability of lambs to progress from standing to suckling across the range of cold stress conditions that we had in this specific data set. And there are some genetic variation in neonatal lamb behavior, but when we look at the literature, the limited literature that is available, there was quite some variation. And then also the latency from standing to suckling was affected by the C-square, or the common maternal environment, and this is related to significant genetic variation when we model this cooperation as a trait of the U that was demonstrated in previous studies. So I think that further research may be required to better understand the response to cold stress in lamb survival. And one of the things that we're doing, and Niels is actually working on it right now, is to apply a reaction norms approach to U to model lamb survival of sire progeny across this whole cold stress trajectory. So we expect to have some results quite soon. And this is it, Chair. There's some people to thank, and that's it.
Thank you for the presentation. Are there any questions for Skalk? Any other question? While we are still there, bringing the mic.
Skalk, thanks. Just a quick question. What do you think the contribution of the lamb's transition from, let's say, an environment of 38.5 to, let's say, 7 degrees Celsius have on the adrenaline profile? Don't you think that might have contributed to the lamb standing up sooner because of that stimulus of the transition from the relative hot to the colder environment?
That could possibly be true. I actually haven't thought about that. Is that the let's say the transition from body temperature to a cold environment would be much greater. So it's a good point. I think so.
And
thanks for raising it.
Yeah, and it would be great also because I've probably contributed to some of that data, but
yeah, you actually also observed some of the lambs.
Maybe also the maybe precipitation, how wet the grass was, maybe as well.
Yeah, I'm actually not sure of that. It's actually quite complex. What we use is we use the temperature data for the day the lamb was born, and what. Because it's spread over a 24-hour day, it could not be precisely at the moment the lamb was born. So despite this, we still get these fairly decent results, telling us that we do have to take cold stress into consideration. Yeah. So I think from that perspective, I'm quite satisfied with the outcome of the study.
Yeah, but I think the adrenaline probably contributed to that quicker standing up.
Yeah, could be.
More question? Okay, that brings us to the end of the session. Thank you, everyone. Let's give ourselves a round of applause. Let's thank all the speakers, especially for making my life easier in observed time where I had to raise my iron fist. Apologies. Unfortunately, the venue is booked for another event. Thank you, Scott. Yeah. Yeah, there's no.
Hi guys, just a quick announcement from VetLink. If you can all please hand in your lanyards before you leave to Madeleine at the desk. We are all about recycling.
Okay. This is what, so all everyone's got on it. Okay. The one left
Okay, can we settle down, guys, so we can continue and you can have lunch? We're gonna be very quick. It's just a few awards that we're gonna hand out, and then we're gonna close maybe with our president just giving us the closing remarks. Before we give out the awards, can I just thank a few people? Obviously, the SASAS Council. I would like to thank them. We'd like to thank all sponsors as well again. I think 3 of them are actually handing out awards now, so that's good. And really, I would like to thank VetLink as well. I think they've been great assisting us, as well as Ascot In for accommodating us and tolerating some of the things we weren't happy about. And then I'll thank my committee after the awards. So can I call Prof. Scott is already here to award the Livestock Improvement Award.
Thank you very much. Good afternoon, ladies and gentlemen. Since last year, the Livestock Improvement Schemes Trust decided to give prizes for posters on animal breeding and genetics only for masters and PhD students. So other people don't qualify for the awards. There's a third prize, a second prize, and the first prize. They are linked to financial incentives as well. The third prize is 500 Rand, second prize 1,000 Rand, and first prize 2,000 Rand. And you will receive an email from the trust asking for you for your banking details. So it's not a scam if they ask you for your banking details. Okay, I won't keep you long. And the adjudication was done by the organizing committee, so I was not involved in it at all. The first prize, the title of the presentation is Genetic Characterization of Non-Descriptive Indigenous Goat Genotypes from South Africa Rural Communities. Ms. Magoro, are you here? It seems she disappeared. Okay. Bad luck. No, she presented. I don't know what happened to her. Okay, we'll keep a certificate. The second prize go. The title is Evaluation of Bull Breeding Soundness and Cow Fertility in South African Smallholder Dairy Herds using Conformation and Physiological Analysis. Buvula, Ms. Buvula? No, I don't know what happened to the students. I know the winner is here. And I think the winner was actually a very deserving candidate because the title is Genetic and Environmental Parameters for Early Life Weight and Tail Length in Merinos. And it was felt that is so important, especially in the with the issue of animal welfare, that should be the first prize. It goes to Tiebus and Skulk, will you please come and Matome, come. Matome Madalindi was the first winner, and I'm going to ask him, to hand it over. The name of the second winner will also be put on. Matome, please hand it over. Ms. Bavula is now here, so I can hand over the certificate to her. You'll see you're getting two certificates. The one's got your name on it. If you think we didn't write it neat enough, there's a second one where you can get somebody to write it for you. And so there's congratulations with the second prize. And now the third prize is also. They are playing a game on you. They want first prize, second prize, third prize. Magoro? She's a master student. Now he receives it on behalf of the student. Yeah. Congratulations, you got the third prize for your poster. Okay. Thank you very much.
Okay, can we move on, please? Thank you, Prof. Scott. Okay, the next award is gonna be for the best nutrition student poster. I'll ask Bonita from AFMA to come and present that one.
Hello everyone. The AFMA Nutrition or the AFMA Student Nutrition Poster Award is presented annually to recognize excellence amongst animal nutrition graduates in their dissemination of nutritional knowledge through a poster presentation at an Animal Science Congress, which is the SASAS Congress this year. The award is designed to promote interest for industry-related research in animal nutrition, to enhance the quality of scientific poster presentations, and to acknowledge excellence in this field. The overall winner will receive a cash prize to the value of 3,000 Rand, as well as a complimentary ticket to our AFMA Symposium this year, which will take place on the 18th and 19th of October. The winner will also receive special recognition of this achievement through acknowledgement in our AFMA Matrix, our electronic AFMA newsletter, as well as our social media. And then the first and second runner-up will also receive acknowledgement and also complimentary tickets to our AFMA Symposium. So the The posters was submitted to us by Trevor, and we have a scientific panel that comprises of members of the industry who evaluates and scores these posters. And I'm going to go, the second runner-up is from the University of Pretoria, Mr. Kubeko Makalima, who presented his poster. So, yeah, he presented his poster on the effect of direct-fed microbial products on performance and intestinal histology in broiler-fed maize, soybean, meal-based diets. So, congratulations. Sorry. The first runner-up is from Stellenbosch University. Unfortunately, she could not make it today, so Hilette will receive on behalf of Ms. Josephine Elizabeth Sillier, who presented a poster predicting the voluntary feed intake of South African boer goat castrates and does from weaning to maturity. And then the overall winner is also from Stellenbosch University. She also unfortunately could not make it today. It's Miss Janica de Beer. She submitted a poster on the effect of decreased concentrate supplementation on milk production and body condition of pasture-fed lactating Jersey cows in a summer grazing cycle. Okay.
Thank you, Vanessa. Okay, our last award for today is from RPO. Unfortunately, Angus is not here, so I'll hand it over. This one was for the best applied animal science student poster, and it goes to Elri Potter. It goes with a 2,000 Rand prize. Okay, just to conclude, I'm going to ask Trevor just to come up after this. But before that, can I please ask all our SASAS committee members to come up front? These ladies and gentlemen are the guys who we've been working with behind the scenes. You might have chatted to them during the Congress. I'd just like to thank them personally, I think, for a job well done. I think we had one of the things that made our work easier, we had a right mix of people. We had the old, we had the young. So we managed to overcome any situation that we got through, And what I enjoyed most working with them was that we debated everything, and we were a team, if I can call it that, and that's what made it easy. So from me, we've got a few gifts we'd like to give to all of you. Thank you. Shift to that side. I think Trevor will take over from here. Thank you.
Thank you, Sibo. Hey, don't disappear. Sibo, don't disappear. Right, thank you. That's coming to the end now. Lunch is waiting for us. Madeleine from Betting and Tina, thank you very much for everything you put in. Madeleine will explain to you now how the app works if you don't know how the app works, because all the information will be there till the end of the year on the website or on the app. Okay.
Just a few important things. We would like to recycle the name tag. So if you can drop it outside if you don't want it, because we use it again. Then for your SACNAP certificate, if you want a certificate of attendance, it is already registered. On the program, there's that QR code. Scan the QR code so that you can get it, and we will send a post-event email and also put it in there. If you put in your particulars, you will receive an email with your certificate attached. And then the last thing, if you want, so if you downloaded the app, when you arrived at the conference, it would have asked you, are you attending virtually or in person? And so it wouldn't show you any of the videos on your app. So to change that, you need to go to the app, to More, and then under More, you will see there's a little setting that says Virtual Event Mode. Just change that, because then you can see the videos. Otherwise, the videos would have made a noise in the room. So that on the website, there's still a link to the Virtual Event app that you can obviously also view on a computer. The code will remain the same, and it will remain available for up to the end of the year or so if you want to go back and watch the sessions that you missed. And then any of the posters that we didn't get in time, we will still go and upload it there. I think that's all. Thanks.
Thank you, Madeleine, and thank you, Tina. You made our life very much easier than it would have been. All right, we've come to the end. It's good to see there's still a hall full of people. Okay. Thank you all for coming. Some of you have come a long way, and we just wish you a safe drive home or flight home, or whichever way you're getting home. And I'm sure you all want to get home, and we hope you'll get there safely. I don't think there's any other thanks. With that, we will declare this Congress closed, have lunch, and on our way. Thank you.